3. Eumycotan fungi, the mainstream and others. Phylum 4 Zygomycota, Phylum 5 Glomeromycota, Phylum 6 Microsporidia. First, a lean item display of the classification to show what goes where. Kingdom Eumycota. Phylum 4 Zygomycota. Subphylum Mucoromycotina, Order Mycorrhizaes. Order Endogonales, Order Mortierelles. Groups currently of uncertain position. Subphylum Entomopteromycotina, Order Entomopterales. Subphylum Zoopagamycotina, Order Zoopagales. Subphylum Kixellomycotina, Order Kixellales, Order Demargaritales, Order Harpalales, Order Acellariales. Kingdom Eumycota. Phylum 5 Glomeromycota. The arbuscular mycorrhizal or endomycorrhizal fungi class Glomeromycetes, formerly placed in the Zygomycota. Eumycotan fungi, the mainstream and others. Kingdom Eumycota, Phylum 6 Microsporidia. Example Nosma, intracellular parasites, mainly of insects, formerly considered protozoa, but now regarded as extremely reduced fungi. C. Carati and Keeling 2009. Rapidly evolving and possibly derived from the Zygomycota. Introduction. Kingdom Eumycota was for many years assumed to be made up of 4 phyla: Chytridiomycota, Zygomycota, Askamycota, and Basidiomycota. These, and particularly the last 2, far outnumber the chromostom pseudofungi in species diversity. We already know about 100,000 Eumycotan fungi, and it is obvious to those of us who work with them that these are just the tip of the iceberg. We estimate that there are well over 1 million species waiting to be found and described. Hundreds of new fungal taxa are described every year. For example, in 1990, in a single publication, Rafael Castaneda and I described 14 new genera and 40 new species of microscopic molds from dead leaves of Cuban rainforest plants. And we have become aware of several new phyla in recent years, as the outline of the phylogenetic system shows. This wealth of species is a measure of fungal success in evolutionary terms, just as the existence of millions of species of insects tells us that they, too, are winners, although their total biomass is far less than that of the fungi. Before we look at the Eumycotan fungi in detail, it is worth inquiring into the reasons for their success. Earlier, I introduced the idea that the number, kind, and arrangement of motility organelles, flagella, found in the chromostom pseudofungi, Eumycota, Hyphocatriomycota, and some Eumycotan fungi, Chytridiomycota, Neocolymastigomycota, Blastocladiomycota, are very basic, highly conserved features. As a corollary of this, the absence of motile cells from the life cycle of most Eumycotan fungi must also be considered important. This seems to reflect a radical shift in evolutionary direction. It shows very clearly that most true fungi are basically terrestrial, and must have been so for a long time, even in geological terms. Many more ecological niches and substrates are available on land than in the water, and the challenges of survival and dispersal are very different. Those true fungi that live in fresh or salt water are secondarily adapted to those habitats, as are, for example, all aquatic mammals. Fungi are heterotrophic, which means that they depend on energy-rich carbon compounds manufactured by other organisms. But this doesn't seem to have been a serious disadvantage. Fungi have evolved enzymes that can digest some extremely tough substrates. Chitin, arthropod exoskeletons, keratin, mammalian and avian skin, hair, horn, and feathers, celluloid, most plant debris, the largest reservoir of biological material, and lignin, a major constituent of wood, nourish many fungi, although we must keep in mind that cellulose and lignin remain completely unavailable to almost all animals, except with the collaboration of microbial symbionts. The unusual ability of some saprobic fungi to exploit cellulose and lignin gives them almost exclusive access to the massive quantities of plant debris produced every year and may well make them the world's #1 recyclers. Only human-made plastics are, perhaps. Chapter 3. Unfortunately, mostly immune to their attacks. This means that we, not the fungi, must take responsibility for recycling these substances. I should add that we are now beginning to develop and use biodegradable plastics, which should somewhat mitigate the problem. There are two major reasons for the incredible success the fungi enjoy. The first is the fungal spore, the second the fungal hypha. Spores permit rapid dispersal and a kind of scattershot saturation of the biosphere. Fungal spores are, almost, everywhere. Hyphae permit the thorough and intimate exploration and exploitation of newly available substrates. Spores. The non-modal microscopic spores of Eumycotan fungi, which come in a dazzling array of forms, Fig 3.1, to fit specific functions, are often produced very 0 I slash equals 1v equals. Fig 3.1 Diversity of spores among Eumycotan fungi. Eumycotan fungi the mainstream and others. Quickly, in a matter of days or even hours after the initial colonization of the substrate. And in enormous numbers. They are dispersed by wind, by water, or by animal vectors, and they can often survive long periods, sometimes even years, of unfavorable conditions such as freezing, starvation, or desiccation, which means drying out and is spelled with one S and two E's. Like bacteria, fungal spores are everywhere. Especially in the soil, in astronomic numbers, and in the air we breathe, sometimes 10,000 or more in a cubic meter, occasionally many more. If you are curious about the ways in which we describe and name these spores, zip off to Chapter 4 and find out. Hyphae. These are the vegetative, assimilative organs of most fungi. When a spore germinates, what emerges is a hypha, sometimes more than one hypha, which grows at its tip and explores the microscopic world in which it landed. The picture below shows hyphae emerging from spores and looking for food. In order to explore their immediate surroundings properly, hyphae must branch repeatedly as they spread out. This is shown in the drawing below. It shows a young fungal colony, Fig 3.2, arising from a single spore, the black dot in the middle. Its strong, waterproof, chitinous hyphae, its richly branched growth pattern, the digestive enzymes it secretes at its growing tips, and the hydrostatic pressures it can bring to bear, all these make it ideally suited for actively penetrating, exploring, and exploiting solid substrates in a manner that the bacteria, chief competitors of the fungi in the recycling business, cannot match. How many hyphal tips do you think there are in this illustration of a very young colony? 176, 294, 388, 502. The answer is 388, and this number will double every hour or so. If a fungus is growing in liquid culture or in a solid substrate and producing a spherical colony, the rate of increase in the number of hyphal tips is much faster, and the final number in a mature colony, astronomical. The transmission electron micrograph, Fig 3.3, from Cole and Sampson 1979, shows how vesicles, V, containing new wall material or enzymes concentrate at the Fig 3.2 young colony of Phycomyces arising from a metospore. Note the large number of hyphal tips and the absence of septa that is typical of Mycorrhizaes. Chapter 3. Fig 3.3 Transmission electron micrograph from Cole and Sampson 1979. Hyphal tip, while the energy engines of the cell, the mitochondria, M, sit a little farther back. The many little round black dots in clusters are ribosomes, the sites of protein synthesis. It is at the hyphal tip that the fungus interacts with the world outside, and where all growth in length takes place. Fungi have learned to cope with environmental extremes. They can grow at temperatures as low as -5 degrees Celsius and as high as 60 degrees Celsius. They include the most zero-tolerant organisms known. Some molds will grow at the amazingly low water activity of 0.65, most plants wilt permanently at a water activity of 0.98. Other molds grow in oxygen concentrations as low as 0.2%, air now contains 20% oxygen. Certain fungi can grow under extremely acid conditions, pH-L, others can tolerate alkalinity up to pH 9. These topics are covered in more detail in Chapter 9, Fungal Physiology and Metabolism, and Chapter 20, Food Spoilage by Fungi and How to Prevent It. As I have already noted, the saprobic fungi are recyclers par excellence, but they are also among the world's greatest opportunists and don't restrict their attentions to naturally occurring dead wood and leaves. Wherever there is a trace of moisture, their omnipresent spores will germinate, and the hyphae arising from them will attack food and fabric, paper and paint, or almost any other kind of organic matter. Some of their metabolites, mycotoxins, are extremely dangerous, even carcinogenic if they contaminate food. Chapter 21, Mycotoxins in Food and Feed. And parasitic fungi cause the majority of serious plant diseases. Chapter 12, Fungal diseases of crops and trees, as well as some of animals and people. Chapter 23, Medical Mycology. Fortunately, there is a brighter side to fungal intervention in human affairs. We have harnessed the biochemical virtuosity of the saprobic fungi in the production of beer, wine, bread, some gourmet cheeses, soy sauce, some antibiotics and immunosuppressants, organic acids, and many other useful chemicals. Fungi are even being used to convert plant waste into high-protein animal feed and into packing materials. We ourselves eat a number of the large, spore-producing structures developed by fungi. Mushrooms, chanterelles, morels, and truffles are all familiar to devotees of French cuisine. Mycotan fungi, the mainstream, and others who prize them for their unique flavors. Some of the parasitic forms are now being recruited to attack insects, weeds, and other fungi which threaten our welfare. Chapter 14. And fungi in intimate, obligatory association with the roots of almost all higher plants, forming mycorrhizas, silently, invisibly, and influentially perpetuate one of the world's oldest and most successful forms of mutualistic symbiosis. Chapter 17. Now for a few more details. Phylum Ascomycota. This phylum is the least well defined at present, since several of the groups traditionally placed in it are now of uncertain disposition, and it is not regarded as monophyletic. Although the subphylum Mycorrhizaes contains only about 1% of the known species of fungi, its members are distinctive, and some of them are common, successful saprobes, fast-growing, primary colonizers of substrates containing accessible carbon sources like sugar or starch. Zygosporangia. The name of the phylum is derived from the way in which its members reproduce sexually by the physical blending, fusion or conjugation of morphologically similar gametangia to form a zygosporangium, the teleomorphic phase. Zygos is Greek for a yolk or joining. The gametangia arise from hyphae of a single mycelium in homothallic species or from different but sexually compatible mycelia in heterothallic species. Zygosporangia usually develop thick walls and act as resting spores. The four diagrams in Fig 3.4 show how a zygosporangium develops. When compatible, mycelia of Phycomyces blakesleyanus meet, individual hyphae establish intimate contact, developing finger-like outgrowths and giving the appearance of grappling with one another. This lets them exchange chemical signals which establish that they are indeed sexually compatible. Then the two hyphae grow apart again, only to loop back, swelling as they approach each other, and finally meeting head-on. They have become gametangia, which fuse when their tips touch. Note that there isn't any sexual differentiation in size or shape here, since we can't call them male and female, we simply label the mycelia plus N. After the walls between the two gametangial tips have broken down and their multinucleate contents have mixed, the mixture is quickly isolated by two septa, one at each side, and the paired-off nuclei fuse. The structure is now called a zygosporangium. and it develops a thick and often ornamented wall, even while still supported on either side by the former gametangia, which are now called suspensors. Although the two suspensors are now just empty appendages, they make it easy to recognize a zygosporangium. When you see one Anamorphs, you won't often see zygosporangia in field collections, although I sometimes find a homophthalic species of syzygites producing them profusely as it parasitizes over mature wild mushrooms. But asexual or anamorphic phases of zygomycetes are easy to find on moldy bread or peaches or on horse dung and dog poop. A number of examples, including a couple of teleomorphs, are illustrated in Figs 3.5 and 3.6. Chapter 3. Fig 3.4 Development of Zygosporangium, Teleomorph, in Phycomyces blakesleyanus, Mycorrhizaes. H. Mainstream and others. Eumycotan fung 1 te. Sporangium columella Sporangium collapsed columella. 3 Spored sporangioles. I. Sporangioles. B. Blake/Ea trispora. A. Rhizopistolonifer. 1 Spored sporangioles. I. Gans. D. Tamidium E. C. Cunm. Gamella. Pseudophylides. 1/1 I. Z. Sporangioles. I. Ecoemancia. F. Spirodactylon. Fig 3.5 Anamorph phs in the Zygomycota. AD Mycorrhizaes, E. F. Kixilils. Chapter 3. Zygosporangium C. Sporangiophore fly exoskeleton D. Zygosporangium hyphal bodies E. F. Fig 3.6 Pylobolus, Mycorrhizaes. A. Habit of sporangiophores, anamorph, on dung, beaded appearance is caused by condensation droplets. B. C. Action of subsporangial vesicle and retinal area in phototroism. D. Zygosporangium, teleomorph. E. F. Entomoptera. Entomopterales. E. Section through the anamorph sporulating on a fly. F. Zygosporangium, teleomorph, arising by fusion of hyphal bodies. Collect some fresh horse dung, from a horse eating naturally in a field, keep it in a damp chamber, and look at it through a dissecting microscope, or even a hand lens, every day. You should be able to follow a sequence of specialized coprophilous fungi, and the first to develop will probably be the spectacular anamorph of Pylobolus, Fig 3.6 AC, which is discussed below and in Chapter 11. The non-modal asexual metospores are usually formed inside mitosporangia borne at the tips of specialized sporangiophores. Zygomyceta cell walls are mainly of chitin, and the nuclei in their somatic hyphae are haploid. Now for a brief taxonomic survey of the phylum. Subphylum 1 Mucoromycotina. I will introduce you to 3 orders, the Mycorrhizaes, Mortierelles, and Endogonales. 1. Order Mycorrhizaes. This order of about 300 species, most of which grow readily and rapidly in culture, includes all the common saprobic zygomycetes. Here belong the ubiquitous bread mold, Rhizopistolonifer, Fig 3.sa, and the almost equally common T. Underscore dot. Underscore comma dot underscore. T. Underscore dot. Underscore comma dot underscore. Uranicotan fungi, the mainstream and others. Genus Mucor. Each globose mitosporangium of these fungi contains hundreds of non-modal, asexual spores, and these sporangia are produced at the ends of tall, stout, simple, or branched hyphae called sporangiophores. The trademark of this group is a swollen extension of the sporangiophore called a columella, Fig 3.sa, d, which protrudes into the sporangium and often persists after the delicate outer skin or peridium of the sporangium has disappeared and the sporangiospores have been dispersed. Other groups often have fewer spores per sporangium, and their sporangia have no columella. Tamnidium elegans, Fig 3.5d, seems to compromise. Its tall sporangiophores have one large, terminal, columellate sporangium, but lower on the stalk there are branches which fork repeatedly in a dichotomous manner, the final branchlets ending in tiny mitosporangia, sporangioles, which contain only a few spores. The reductionist tendency is also evident in Helicostylum, which has 10 to 20 spores per sporangium, and in Blakesley Trispora, Fig 3.sb, which has only 3 spores per sporangium. This trend reaches its logical conclusion in Cunninghamella, Fig 3.sc, which has only 1 spore per mitosporangium, and in which the walls off spore and sporangium appear to have fused. Now the whole mitosporangium becomes detached and acts as a dispersal unit. Although Mycorrhizaes can go through cycle after cycle spore, mycelium, sporangium. Spore producing only the anamorph, they sometimes form sexual zygosporangia. The teleomorph, perhaps as a survival mechanism, perhaps for the benefits of genetic recombination, or perhaps just because compatible strains have met. The photo, Fig 3.7, shows both teleomorph and anamorph of Phycomyces. Underscore comma. Colon. Colon dash C comma colon. Dash, Z comma dot comma. TF. R, colon, comma dash. Dash. And GTT. Dash. Backslash. Comma. Dash. 1, comma colon. Fig 3.7 Teleomorph and Anamorph of Phycomyces. Chapter 3. The anamorph-teleomorph alternation is diagrammed in Fig 3.8 for one of the most common and successful members of the Mycorrhizaes, Rhizopistolonifer. Note that when the zygosporangium germinates, it produces a mitosporangium rather than a germ tube. Somatic hypha germination Me10s1s/sporangi_opheres I /mature with zygosporangium mitosporangia suspensor zygosporangium somatic hypha Fig 3.8. Mycorrhizaes, life cycle of Rhizopistolonifer. Sporangiospores O sporangiospores. 0, J. 0 FJO. E, OTSJ. 00 CL. Germination. Registered L. Rhizoids. Eumycotan fungi the mainstream and others. Zygosporangia vary in minor ways from one genus to another and among families and orders, but they are generally rather similar, Figs 3.4, 3.6d, f, 3.7, 3.8 if they are present, they are the easiest way to tell if a fungus is a zygomycete. By contrast, the anamorphs of zygomycetes mitosporangia and the structures on which they are born have evolved some amazing and bizarre adaptations. This contrast between teleomorphic constancy and anamorphic diversity is presumably the result of differing evolutionary pressures. Long-term survival, one of the main objectives of the teleomorph, is presumably best ensured by structures with minimal surface area and thick, protective walls. Dispersal, the main purpose of anamorphs, can be achieved in many ways. Let's look at 3 of the more specialized zygomycetes anamorphs. Pylobolus crystallinus, Fig 3.6 AC, is an atypical but fascinating coprophilous, dung-inhabiting, member of the order Mycorrhizaes. It grows very rapidly and is one of the first fungi to fruit in the extended succession that occurs on dung. See Chapter 11 for a detailed expose of this succession. Its unbranched sporangiophores are 2 to 4 centimeters tall and have a unique explosive dispersal mechanism. The whole sporangiophore is a single large cell. Beneath the black apical mitosporangium is a lens-like subsporangial vesicle, with a light-sensitive retina at its base that controls the growth of the sporangiophore very precisely, Fig 3.6 BC, aiming it accurately toward any light source. In a word, it is phototropic. The generation of osmotically active compounds inside this giant cell causes hydrostatic pressure in the sporangiophore and the subsporangial vesicle to build up until it is over 100 pounds per square inch, 7 kg per square cm. This eventually causes the vesicle to explode, hurling the black sporangium away to a distance of up to 2 meters, directly toward the light. The mucilaginous contents of the subsporangial vesicle go with the sporangium and glue it to whatever it lands on. Can you explain why Pylobolus needs such a specialized mechanism for spore dispersal, such a powerful cannon, so carefully aimed? You can find the answer in Chapter 11. Note that the originality of Pylobolus extends only to the behavior of its anamorph. Teleomorph, the zygosporangium, shown in Fig 3.6d, is fairly conventional. 2. Order Mortierelles, almost 100 species in 6 genera, but 90 of those species in Mortierella. The zygosporangia of these fungi resemble those of the Mycorrhizaes, but the asexual sporangia do not have a columella. 3. Order Endogonales, 4 genera. These fungi are among the relatively few that establish mycorrhizal symbioses with plants. Some produce pea-sized sporocarps that contain numerous zygosporangia, see www.mycolog.com. Subphylum 2 Crystallinus. This contains the orders Cyxalales, Demargaritales, Harpalales, and Acellariales. This seems rather strange because members of the first order are largely coprophilous saprobes, the second order is parasitic, while the last two are biotrophic commensals inside the guts of living insects and have been traditionally separated as the class Trichomycetes. But see note below defining this group in an ecological rather than phylogenetic way. Chapter 3. 1. Order Cyxalales, named after a mycologist called Cyx. Members of this order are atypical of the zygomycetes in that they often have regularly septate hyphae. Their teleomorphs are unremarkable, but they develop some oft-most complex anamorphs. Known, Fig 3.se, f. I have found Coemantia, Fig 3.se, on bat dung from a cave. Its tall sporangiophore bears many fertile side branches called sporocladia. Each of these produces a row of lateral cells called pseudophyalides. True phyalides are discussed in Chapter 4. Finally, from the apex of each pseudophyalide arises an elongate, one-spored mitosporangium, often called a sporangiole. That's complex enough, but it looks simple beside Spirodactylon, Fig 3.sf. This, surely the most elaborate of all zygomycetes anamorphs, produces a tall, branched sporangiophore that is repeatedly thrown into tight coils. Within these coils arise the sporocladia, which bear pseudophyalides, and these in turn bear one-spored sporangioles. It is hard to imagine why this strange configuration might have evolved, until one learns that the fungus grows on mouse and rat dung. Coprophilous fungi have various highly evolved strategies for getting back inside the gut of the animals that produce their preferred substrate. This isn't too difficult for genera like Pylobolus, which grow on herbivore dung, since all they have to do is get their spores onto the animal's food, which is all around. But rats and mice are omnivores, not herbivores, and it is essentially impossible for the fungus to ensure that its spores will be present on their food. The only alternative, as I see it, is to attach spores to the animal itself in the hope that they will be ingested during grooming activities. Rats and mice are creatures of habit, using well-trodden paths each day. Along these trails they deposit dung, and there, later, the coils of Spirodactylon become entangled in their hair. Only the zygosporangia of the Cyxalales convince us that these strange fungi are indeed zygomycetes. 2. Orders Harpalales and Acellariales. These eccentric groups of fungi live almost exclusively attached to the lining of the guts of living arthropods, which is why you won't run into them very often, if at all. But they are examples of the opportunism displayed by fungi and the determination shown by mycologists in winkling out fungi wherever they are to be found. Bob Lichtward's 1986 book gives a fine account of these offbeat groups. A plate, Fig 3.9, of phase contrast photomicrographs by Richard Benjamin, 1979, shows the characteristic structures of some Trichomycetes. Top left, A, are developing trichospores of Smydium. Top right, B, are trichospores of Stachylina showing the hair-like appendages that give them their name. These are not to be confused with flagella. Bottom left, C, is a trichospore of Smydium. Bottom center, D, is a developing zygospore of Trichozygospora, and bottom right, E, a released, mature zygosporangium. With a collar and a bunch of hair-like appendages below it. This last structure, look at the conical suspensors, top and bottom, places these Trichomycetes in phylum Zygomycota. I must mention here that the Trichomycetes are now regarded as a polyphyletic group defined in ecological terms. In addition to the two orders mentioned above, there are two groups often called Trichomycetes that are actually protozoans, the orders Amoebidales and the Ecrinales, which occur, just as the Harpalils and Acellarials do, as commensals attached to the gut walls of living invertebrates. Eumycotan fungi, the mainstream and others. Fig 3.9 Phase contrast photomicrographs of Trichomycetes. Chapter 3. Subphylum 3 Entomopteromycotina. 1. Order Entomopterales. As the name implies, these fungi often attack insects. Entomoptera muscae infects, and eventually kills, houseflies. Dying flies, their bodies riddled by the fungus, usually crawl into exposed situations. I find them on windows and on the growing tips of shrubs in my wife's garden, where the fungal infection bursts through the exoskeleton and produces tightly packed masses of sporangiophores, Fig 3.6e. Each sporangiophore bears one unicellular, sticky mitosporangium that is shot away at maturity. When the fly dies on a window, this barrage produces a whitish halo of mitosporangia on the glass. These sporangia can infect other unsuspecting flies that come to pay their last respects. As you may already have guessed, species of Entomoptera are being investigated for their potential use in biological control of insect pests, see Chapter 14. Note again that the zygosporangia of Entomoptera, although developing in an unusual way, by the fusion of hyphal bodies inside the fly, are still recognizable as zygosporangia, Fig 3.6f. Basidiobulus and Candidiobolus cause serious animal, including human, diseases. Subphylum 4 Zooapagamycotina. Order Zoopagales. Most of the almost 200 species of this inconspicuous order are parasites or predators of microscopic organisms, such as Amoebae. Phylum 5 Glomeromycota. Endomycorrhizal or arbuscular mycorrhizal fungi. 5 orders, 29 genera, about 230 species. These fungi are dealt with in some detail in Chapter 17, because they are involved in more mycorrhizal relationships with plants than any other group. The soil-inhabiting Glomeromycota were placed in the Zygomycota only tentatively, since the only genus that develops zygosporangia is now known to belong elsewhere. Nevertheless, they are extremely important fungi, because their hyphae enter the living root cells of perhaps 90% of all higher plants and establish with them obligate mutualistic symbioses called arbuscular mycorrhizas, AM, or endomycorrhizas. These are illustrated and discussed in Chapter 17, my main treatment of mycorrhizas. AM fungi won't grow in azenic culture, they must be associated with a living plant root. They're generally very large, up to 600 microns, and thick-walled resting spores are common in most soils and are stimulated to germinate by the proximity of plant roots. Almost any plant will do, because these fungi have such wide host ranges. They're usually non-septate hyphae spread through the soil and enter living roots, where they develop diagnostic structures intracellular, finely branched, tree-like arbuscules, Fig 17.3, which are the interface across which the fungus exchanges mineral nutrients, especially phosphorus, for photosynthase, sugars, etc., provided. Eumycotan fungi, the mainstream and others. By the plant. Many of the Glomeromycota produce both arbuscules and lipid-filled structures, called vesicles or intramatrical spores, inside plant roots. The soil-inhabiting mycelium is very efficient at mobilizing insoluble phosphorus and translocating, moving, it and other mineral nutrients to the plant. Since phosphorus is often the limiting nutrient for plant growth, fungi help plants to thrive in poor soils. These fungi are therefore vital in many natural habitats and of great potential value in agriculture. Again, for details, consult Chapter 17. Molecular biology strongly suggests that the Glomeromycota nest with the Askamycota and Basidiomycota. Phylum 6 Microsporidae. These organisms, about 1,500 known species, are all intracellular parasites of animals, mainly insects but also fish, crustaceans, and even humans. They have no mitochondria or flagella and reproduce mostly by forming extremely resistant spores. Nosma is the best known example, and parasitizes many different insects. It is highly unlikely that you will ever see members of this group. Further reading. Benjamin, R.K. 1959. The Merisporangiferous Mycorrhizaes Aliso 4:321-453. 1979 Zygomycetes and their spores in the whole fungus, edited by B. Kendrick, 573-621. Volume 2. Ottawa, National Museums of Canada. Bentavenga, S.P. 1998. Ecology and Evolution of Arbuscular Mycorrhizal Fungi McIlvania 1330-39. Brundret, M., L. Melville, and L. Peterson, eds 1994. Practical Methods in Mycorrhiza Research Mycology Publications, 8727, Lochside Drive, Sydney, BCVSLIMS, Canada CD-ROM. Serta Almedo, E. and E. D. Lepson, eds 1987. Phycomyces. Cold Spring Harbor, New York, Cold Spring Harbor Laboratory Press. Cole, G.T., and R.A. Sampson. 1979 Patterns of Development in Conidial Fungi. London, Pittman Publishing. Carady, N., and P. J. Keeling. 2009 Microsporidae, A Journey Through Radical Taxonomic. Revisions Fungal Biology Reviews 23:1-8. Fuller, M.S., ed. 1978 Lower Fungi in the Laboratory. Athens, University of Georgia Press. Ingold, C.T. 1978. The Biology of Mucor and Its Allies. London, Edward Arnold. Kendrick, B., and S. M. Burke. 1985 Mycorrhizae, Applications in Agriculture and Forestry in Comprehensive Biotechnology, edited by C. Robinson, 109-15. Volume 3. Oxford, Pergamon. Lichtward, R.W. 1986. The Trichomycetes. New York, Springer Verlag. Morton, J.B., and G. L. Benny. 1990 Revised Classification of Arbuscular Mycorrhizal Fungi, Zygomycetes, a new order, Glomales, 2 new suborders, Glomania and Gigasporinea. Chapter 3. And 2 new families, Acolosporaceae and Gigasporaceae, with an emendation of Glomaceae. Mycotoxin 37:471-91. O'Donnell, K.L. 1979. Zygomycetes in Culture. Athens, University of Georgia Press. Zika, H., R. Siepman, and G. Linman. 1969 Mycorrhizaes. In Beschreibung aller Gattogenuende Arten dieser Pilzgruppe. Lehr, Germany, Kramer. 4. Kingdom Eumycota, True Fungi, Subkingdom Dicaria. Phylum 7 Askamycota, the Askamycetes. Introduction. Zygomycetes are terrestrial fungi, there's no doubt about that. But they usually thrive and sporulate only in damp places where the atmosphere is more or less saturated with moisture. For example, Rhizopistolonifer will colonize the moist interior of a loaf of bread but won't produce its characteristic sporangiophores and mitosporangia on the outside of the bread unless the surrounding atmosphere is humid. If we persuade the fungus to sporulate by keeping the loaf in a damp chamber, a plastic bag containing a few drops of water will do, and then take it out of the bag, the sporangiophores will quickly collapse. Most of the fungi covered in this chapter and the next are much less delicate and thrive in or on a much wider range of substrates. Subkingdom Dicorea. Phylum 7 Askamycota Introduction. Also comparing Phylum 8 Basidiomycota, see Chapter 5. Hyphae of most zygomycetes are wide, thin-walled, and cynocytic continuous tubes with no cross walls, as shown in Fig 3.2. Hyphae of subkingdom Dicorea, Phylum Askamycota plus Phylum Basidiomycota, are usually narrower. Hyphae average about 5 microns in width, but in aggregate they are very long, sometimes kilometers per gram of soil. They are also septate. They have cross walls called septa at regular intervals. Fig 5.1. These miniature bulkheads give the hyphae some physical rigidity and limit loss of cytoplasm if the outer hyphal wall is ruptured. As a result, we find that the Dicorea can grow in a wide range of conditions. For example, they can often spread and fruit in drier situations than zygomycetes could tolerate. Some Dicorean anamorphs, especially chylomycetes, those with closed fructifications, grow in dead leaves and stems of desert plants, and some molds are the most drought-tolerant of all. Chapter 4. Organisms, able to grow at water activities below 0.70, for example, on jams, saltfish, and other substrates of extremely high osmotic pressure, see Chapter 20. While many zygomycetes can assimilate only accessible substrates like sugars and starch, Askamycetes can often exploit cellulose, and many basidiomycetes can digest both cellulose and lignin, carbon sources that are available to remarkably few other organisms. Although fungi cannot fix atmospheric nitrogen, this talent is restricted to some prokaryotes. Dicorean fungi can use many different forms of combined nitrogen. Some Askamycetes even specialize in metabolizing the protein keratin, which is the main component of hair, skin, and nails. In case you were wondering if those fungi might constitute a health hazard, some of them do. Some other orders of Askamycetes are obligate parasites of plants. Remember the downy mildews caused by Mycota? There are also plant diseases called powdery mildews that are caused by Askamycota. The similarity of common names is unfortunate. But try to remember the difference, because although the groups of fungi involved are both obligately biotrophic, the diseases are different in many important ways, such as host ranges and methods of control. This is just one example of how taxonomy has practical implications, see Chapter 12. Thousands of basidiomycetes, and quite a few Askamycetes, establish intimate mutualistic symbioses, Mycorrhizaes, with the roots of trees, especially conifers, see Chapter 17. Nearly 20,000 Askamycetes, and a few basidiomycetes, have domesticated algae and or cyanobacteria, thus becoming lichens, which can live in some of the world's harshest climates and colonize the barest and most inhospitable substrates, see Chapter 7. Some Dicorean fungi have even re-entered the water and, lacking motile cells, have evolved other mechanisms, such as long appendages, to aid spore dispersal. Dicorean fungi range from unspecialized, almost omnivorous saprobes to fungi so specialized and ecologically demanding that they are found only on one particular leg of one species of insect. Some Dicorean fruit bodies are microscopic, as in most Askamycetes. But often, especially among the basidiomycetes, they are large and complex, and most of the common names applied to fungi refer to the visible teleomorphs of basidiomycetes, mushrooms, puffballs, bracket fungi, and, in a few cases, Askamycetes, morels, cup fungi. You may already be acquainted with some of these. I will introduce you to many more in the pages ahead. Characteristics of teleomorphs. Most Dicorian fungi share a number of important features: 1. Chitinous cell walls, 2. Hyphae with regular cross walls called septa, centrally perforated to allow movement of cytoplasm, and sometimes nuclei, between compartments, 3. The ability of somatic, assimilative hyphae to fuse with one another, anastomosis, and to exchange nuclei, and 4. The occurrence in their life cycles, or at least in those which produce a teleomorph, of a unique nuclear phenomenon called the Dicorion. After sexually compatible nuclei from different mycelia have been brought together by anastomosis, they pair off but do not fuse immediately to form a diploid zygote. Instead, they go on dividing synchronously to populate the cells of what are called dikaryotic hyphae, in which each cell or compartment has two sexually compatible haploid nuclei. Oh yes, they do fuse eventually, but not before some remarkable developments have taken place. In Kingdom Eumycota, true fungi, subkingdom Dicorea, Basidiomycetes, that ultimate sexual fusion may be delayed for years, and the number of fusions multiplied enormously. If Askamycetes and Basidiomycetes share all these things, how do they differ? Actually, in many ways, and with experience it's usually easy to tell their sexual fructifications apart with the naked eye. But their microscopic, unicellular myosporangia are most diagnostic of all. A8 endogenous myospores ascus. Clamp connection meiosis. 4 exogenous. Hashtag. Myospores. Basidium. V. B. 1. I backslash. I. Fig.4.1 Comparison of Ascus and Basidium development. The myosporangia of Askamycetes are asci, singular, ascus. They are cylindrical or sac-like and at maturity usually contain 8 haploid spores, ascospores, which are in most cases expelled into the air through the top of the ascus. The myosporangia of Basidiomycetes are basidia, singular, basidium. They usually have 4 tiny projections called sterigmata, each bearing a haploid spore, basidiospore, which is shot away individually at maturity. The formation of asci or basidia marks the end of the Dicorea phase. The paired nuclei have fused, and the resulting zygote has undergone meiosis, and a subsequent mitosis in Askamycetes, to produce 8 haploid ascospores or 4 haploid basidiospores. Compare the two sets of diagrams in Fig 4.1, and note how similar the developmental processes are until the final stages. Chapter 4. Teleomorph stage of Askamycota. Now let's examine the sexual, teleomorphic, part of the Askamycete life cycle from the beginning. When an ascospore germinates, it establishes a haploid mycelium. In heterothallic Askamycetes, this can't undergo sexual reproduction until it meets another compatible haploid mycelium. When this relatively rare event takes place, the fungus cleverly maximizes the ensuing potential for genetic recombination in a unique way. One would expect a single sexual fusion, resulting in a single zygote. But most Askamycetes interpolate a Dicorea phase, during which the number of pairs of compatible nuclei is multiplied, often enormously, as dikaryotic hyphae, often called ascogenous hyphae, as in Fig 4.2, grow and branch within a mass of monokaryotic, haploid, tissue, which is the framework of the fruit body, the ascoma. Eventually, the ultimate branches of the dikaryotic, ascogenous, hyphae, of which there will be millions in larger cup fungi, reach their ordained positions in the future hymenium and the long-delayed sexual fusions take place in cells called asci. The genome is reshuffled during the ensuing meiosis in each ascus. This genetic recombination is due to crossing over, which is explained in Chapter 10. Each meiosis, having different patterns of crossing over, will produce a somewhat different arrangement of the genome. In this way the products of a single hyphal encounter are first multiplied, then, on top of that, the reshuffling of genes inherent in meiosis generates a lot of genetic diversity. Not only is the dicryon itself an unusual phenomenon, but during the dicryo phase an effectively diploid mycelium is growing within, and drawing nourishment from, the haploid ascoma tissue. This phenomenon has interesting parallels in the red algae, although molecular evidence doesn't support a close relationship between the two groups. Both lack modal gametes and appear to have simply hit upon the same solution to the problem posed by the rarity of sexual encounters. Ascospores are not modal, in the sense of self-propelling, but most Askamycetes nevertheless send their ascospores off with a burst of kinetic energy. Most asci are tiny spore guns, which work by building up internal pressure then releasing it through the tip. The job of most asci is to get their ascospores into the turbulent airflow above the ascoma. Mature asci of the dung-inhabiting Ascobolus, Fig 4.2, project above the hymenium and point toward the light before discharging their spores. In this way they ensure that the spores will not run into any obstacles on their upward flight, see Chapter 8. 4 kinds of ascoma. The multicellular structures, ascomata, that produce the asci, and act as the platforms from which the spores are launched, come in 4 main designs, sectional views of which are drawn in Fig 4.3 and photomicrographs given in Fig 4.4. 1. Apothecial ascomata allow many asci to discharge simultaneously because the entire fertile layer or hymenium is exposed. 2. Parathecial ascomata have a narrow opening which permits discharge of the contents of only one ascus, or a few asci, at a time, as do 3. Pseudothecial ascomata, similar type of discharge, but different development. Different bitunicate asci. Kingdom Eumycota, true fungi, subkingdom Dicrya. Anastomosis. 0. O. V, dash comma backslash ascogonium. Dot comma dash dash, er. Ascogonium I, O. O slash HL and LT. I. PF hashtag. Underscore F. W antheridium. RR backslash. 2 backslash. II. Ascogenous hyphae. Ascospores. J. T. Backslash. 0. Phototropic asci. R. Backslash slash 0. vs apothecial ascoma. Fig 4.2 Teleomorphic cycle of an apothecial ascomycete, Ascobolus, Pylobolus, see text for full explanation. 4. Cleistothecial ascomata lack an opening entirely. This usually indicates that the asci are spherical, as in these illustrations, and no longer shoot their spores. The fungus has evolved another dispersal strategy. That may have happened because the fungus fruits in a confined space, for example, under bark or below the surface of the ground, where airborne dispersal cannot operate. We often find that the spores of such fungi are dispersed by animals, usually because the fungus has evolved chemical attractants. Chapter 4. Operculum apical ring. Lo. 3. 2. A. Asca 4. Of a yeast. Apothecial ascoma. Unitunicate asci. Osteole prototunicate ascus. Cleistothecial ascoma. Thick inner wall. Thin outer. Pseudoparaphyses wall. Bitunicate ascus jack in the bucks. Fig 4.3 Teleomorphs, asci, asca 4, and sectional views of ascomata. 4 kinds of ascus. Before we go on to explore the many orders of Askamycota, we must take a closer look at the ascus itself. All asci are not the same. There are 4 flavors, Figs 4.3, 4.5. 1. Unitunicate operculate asci, which have a single functional wall with a built-in lid or operculum at the tip. At maturity this pops open so that the spores can be ejected. Unitunicate operculate asci are found only in apothecial ascomata. 2. Unitunicate inoperculate asci, which have no operculum but have a special elastic ring mechanism built into their tip. This is a preset pressure release valve, or sphincter, and the ring eventually stretches momentarily, or turns inside out, to let the perithecial ascoma, pseudothecial ascoma, kingdom Mycota, true fungi, subkingdom Dicaria, A, B, C, D. Fig 4.4. A. Apothecial ascomata, B. Perithecial ascoma, C. Pseudothecial ascoma in vertical section, D. Cleistothecial ascoma. Spores shoot through. Such inoperculate asci are found in perithecial and some apothecial ascomata. 3. Prototunicate asci, which have no active spore-shooting mechanism. These asci are usually more or less spherical and are most often found in cleistothecial, closed, and hypogynous, underground, ascomata. Sometimes the wall of this kind of ascus dissolves at maturity and releases the ascospores, which can then ooze, rather than be shot, out of the ascoma, or they may wait inside until it decays or is ruptured. These asci are often called prototunicate. Yet perhaps because they are found in several otherwise rather different orders, it seems likely that they represent a secondary condition. Chapter 4. A, B, C, D, E. Fig 4.55 Photomicrographs. A. Operculate asci of Pezisa, with the opercula stained blue. B. Asci and ascospores of Colossypha. C. Bitunica with pigmented dictyoseptate ascospores. D. Transmission. Electron micrograph of an operculate ascus showing vertical section of sphincter. E. Prototunicate asci of tuber californicum. Kingdom Mycota, true fungi, subkingdom Dicaria, and have evolved several times from unitunicate asci, as they clearly did in the truffles. See Tuberaceae later in this chapter. 4. Bitunicate asci, which have a double wall. A thin, inextensible outer wall covers a thick, elastic inner wall. At maturity the thin outer wall splits, and the thick inner wall absorbs water and expands upward, carrying the ascospores with it. This jack-in-the-box design allows the ascus to stretch up into the neck of the ascoma to expel its spores. The bitunicate ascus, although still a spore-shooting device, is so different from the unitunicate ascus that they clearly diverged a long time ago. In many unitunicate Askamycetes, the perithecial ascoma develops only after the sexual stimulus, so that the asci can grow into an actively enlarging cavity. In many bitunicate Askamycetes, fertilization doesn't happen until after a solid primordium or stroma has developed, so room has to be made for the asci by dissolving away existing tissue. In some cases the asci themselves do the job, but in others it is carried out by special sterile hyphae, pseudoparaphyses, growing down from the upper layer of the stroma. The asci then grow up between them. Remember that ascomata which develop in this way, pseudothecial ascomata, always produce bitunicate asci. Now for the other half of the story. Anamorph stages of Askamycota. Here is a mantra to begin with, say it until you know it. Holomorph equals anamorph plus teleomorph. The whole fungus equals asexual reproduction plus sexual reproduction. In any modern consideration of the Askamycetes, we cannot ignore their asexual reproductive phases, many of which are called molds, Hyphamicites and Chylomycetes. You already know that Zygomycota have diverse asexual phases, often also called molds. So you won't be surprised to discover that many Askamycetes have comparable asexual, anamorphic, phases during which they reproduce rapidly, and often relatively cheaply, by means of metospores called canidia, which are often just modified bits of hyphae. The asexually reproducing phase of the Askamycete life cycle was more or less ignored for many years in favor of teleomorph studies. But when we consider that the anamorph is an important, and sometimes the only, phenotypic expression of many Askamycotan genotypes, we realize that it has much to tell us. Besides, One can get DNA and RNA from Anamorphs just as easily as from Teleomorphs, so we are beginning to understand the relationships and taxonomic dispositions of Anamorphs much better, even in many cases where no Teleomorph is known. Although they play essentially the same role in the life cycle, the Anamorphs of Askamycetes differ from those of Zygomycetes in two very important respects. 1. While Zygomycetes Metospores commonly originate by free cell formation inside a sporangium, many spores cleaving from a single mass of cytoplasm, the Metospores Canidia, of Askamycetes are basically just modified bits of hyphae, either buddied out as a new structure or converted from a whole existing cell. 2. In Zygomycota, Anamorph and Teleomorph sometimes occur together, especially in homothalic species, and always share the same binomial. In Askamycota. Chapter 4. Anamorph and Teleomorph often develop at different times and on different substrates. Each phase has often been collected in total ignorance of the existence of the other, and because of this, the International Code of Botanical Nomenclature has, until very recently, maintained that it is legal to give them separate binomials, a useful option but one giving rise to a great deal of confusion in some students' minds. How can one organism have two names? Recently, this statute has been overturned, but we now have to decide which name takes precedence, and the idea of calling a well-known sexual fungus by the name of its asexual phase, just because the latter was described first, perturbs many experienced mycologists. It will be a while before things settle down. Several thousand Anamorph-Teleomorph connections have now been established in the Askamycetes. Known connections represent only a small proportion of the total number of taxa, either of Anamorphs or Teleomorphs. We know about 30,000 of each. Because Anamorphs so often occur alone, as mentioned above, it is still common practice to use separate names for them, as you will see, although the latest code of nomenclature specifies that there can be only one binomial per species, a decision with many as yet undetermined consequences, since sometimes the Anamorph was described first, while in other cases the Teleomorph was seen first. I have always found it completely unacceptable to talk about conidial fungi, like many other texts do, as if they constitute a separate major high-level taxon called the Deuteromycotina. This ignores both the evidence that they are all expressions of Dickerean genomes and the thousands of connections that have already been established with Teleomorphs, a number which grows every year. The word Deuteromycotina should be excised from the mycological lexicon. I advocated this in a paper many years ago, Kendrick 1989. See references. In addition, we no longer call the anamorphic fungi imperfecti. Of about 30,000 known Askamycetes, about 5,000 have so far been connected to their anamorphs. What about the many thousands of conidial, anamorphic, fungi that are still orphans? I think there is good reason to believe that many of them have given up sex altogether, and become anamorphic holomorphs, although they seem to have retained some genetic flexibility by, a. having more than one kind of nucleus in their mycelia, heterokaryosis, as a result of occasional hyphal fusions, anastomoses, and, b. sometimes undergoing a complex parasexual process involving rare somatic diploidization, mitotic crossing over, and finally a return to the haploid condition. This process is more fully explained in Chapter 10. Molecular biology now allows us to place anamorphs in teleomorphic families and orders, so the confusion is decreasing. It turns out that the conidial fungi are a mixed bag. Although most of them are, or were, part of Askamycete life cycles, others are, or were, connected with Basidiomycetes. Despite this mixed ancestry, we have had to set up a single classification for all of them, because it is often impossible to tell, just by looking at them, in which phylum the connection lies. Unfortunately, our scheme for classifying the anamorphs was formerly unable to make little reference to teleomorphs, for several reasons. 1. Teleomorphs are known for only 10%-15% of anamorphic species. 2. members of what seems to be a single morphologically coherent Anamorph genus may have teleomorphs in many different holomorph genera, even from different orders. This must be due to convergent evolution among Anamorphs, and 3. Anamorphs belonging to several different Anamorph genera can have sexual phases in a single holomorph genus. This kingdom Eumycota, true fungid subkingdom Dicaria, must be due to radiative evolution among Anamorphs. In addition, quite a few teleomorphs have more than one Anamorph. We call such Anamorphs synonymorphs. So our attempts to classify Anamorphs have concentrated on a. Their Metospores, Canidia, b. The diverse structures, Candidiogenesis cells, Candidiophores, and Candidiomata, which bear them, c. The ways in which the Metospores develop, Candidiogenesis, d. The way in which they are liberated, dehiscence, and, more recently, e. Molecular techniques, which are now helping us to elucidate their relationships. Seifert et al. 2011. Morphology of Anamorphs. There are at least 2,000 described genera of conidioid fungi and almost 30,000 described species, and these numbers are increasing rapidly. The first really useful classification of Dicarian Anamorphs, established in the late 19th century, was based on mature morphology. The principal characteristics used were 1. Color, septation, cross walls, and shape of canidia, 2. Canidiophore aggregation or lack of it, and 3. The production of canidia in enclosed structures or the absence of such enclosure. These characteristics are illustrated in Fig 4.6. The division of Anamorphs into two large groups, Hyphamicites and Chylomycetes, is informal, is based on characteristic 3 above, and is largely for convenience, since the easiest decision to make is usually, although not always, whether a conidioid fungus is a hyphamicete or a chylomycete. You can recognize a hyphamicete because its canidiophores can be single or aggregated in various ways but are never enclosed within a covered canidioma. Chylomycetes form their canidia in initially enclosed candidiomata, which usually develop just beneath the surface of their plant substrate. I am not going to discuss chylomycetes in detail, although there are thousands of them, and they cause many important diseases of crop plants, but I must mention a few basic facts. Chylomycetes equals covered or osteolate candidiomata. A covered candidioma is called an acervular candidioma or acervulus, Fig 4.6, bottom row. This may develop at various depths within the host. It can be subcuticular, covered only by the host cuticle, intraepidermal, arising within the cells of the epidermis, as in the diagram, subepidermal, or developing beneath several layers of host cells. Under this roof of host material, fungal hyphae aggregate and form a flat fertile layer, or hymenium, of short canidiophores that produce many canidia, shown in a vertical section. The pressure of accumulating canidia, and often ophaxosory mucilage, eventually splits the host epidermis and allows the canidia to escape. At the other end of the spectrum is the flask-shaped, osteolate pycnidial canidioma, or pycnidium, seen in vertical section, in which the fungus itself provides the enclosing wall and canidia eventually ooze out through a narrow apical pore or ostiole. Between these extremes lies a range of sometimes hard to categorize fructifications that are discussed succinctly by Kendrick and Nagraj, 1979. Browsing the superbly illustrated books by Sutton, 1980, and Nagraj, 1993, will give you an appreciation for this amazing plasticity. 66. Didymoamero. B. Simple. Chapter 4. 1J, 1A. Helicoaceti-stauro. Complex. Penicillate. Dictea. E. Sporido. Enema. Canidiophores. Acervulus. 0. N. \VJ_LJ. L host. Fig 4.6 Anamorphs, Canidia, Canidiophores, and Candidiomata. Hyphamicitis equals exposed Canidiophores or Candidiomata. Framo. F. Scolico. G. Among the Hyphamicites, Canidiophores are usually solitary, although they sometimes form columnar aggregations, called cinematal Candidiomata, or cushion-shaped masses, called sporodicoel Candidiomata, all shown in Fig 4.6. Kingdom Eumycota, true fungi, subkingdom Dicaria. Canidium morphology. Presence or absence of pigment is important, as are shape and septation. There are 7 shape and septation categories. Fig 4.6 top row. A. Commonest of all are syn. Gl amerospores. B. The addition of one cross wall makes them didymospores. C. Canidia curved through more than a half circle, or coiled in 2 or 3 dimensions, are called helicospores. D. Those with several conspicuous, radiating arms or other projections are called staurospores. E. Septa running 2 ways, like the meshes of a net or like the mortar layers o fabric wall, identify dictyospores. F. 2 or more transverse septa, arranged like the rungs of a ladder, characterize phramospores. G. Finally, those which are long and thin, more than 15 times as long as they are wide, are called scolicospores, which means worm-like. Candidiogenesis, metospore development. More recently, it was discovered that conidial fungi use a number of different techniques to produce their spores. Since these often represent genuine embryological differences, they have become important characteristics in our classification. Spores which look alike can develop in different ways. We begin by checking an anamorph to see which of 2 basic patterns of development blastic or thallic exhibits. Fig 4.7a, b. A. Blastic Candidiogenesis. B. Thallic Candidiogenesis. Fig 4.7 Basic modes of canidium development and release. H. C. Schizolytic succession. D. Raxolytic succession. Chapter 4. Blastic acropetal. Blastic synchronous. /1. Gonidobotrium botrytis. I1. R. Manilia 1 cladosporium. -9. Comma. And //JJ. F. Blastic sympodial. 8. II. Bovaria scopulariopsis. Spilocea. Basidimbria. Blastic analytic. Blastic sympodial rhinocladial penissif/em aspergillus. Blastic phyolitic. Leptographium. Fig 4.8 Representative anamorphs with blastic candidiogenesis. In blastic candidiogenesis, Fig 4.7a, the young canidium is recognizable before it is cut off by a cross wall. This is an extension of the idea of cells budding. In thallic candidiogenesis, Fig 4.7b, the cross wall is laid down before differentiation of the canidium begins. Ripe canidium may also be liberated in 2 different ways, described as schizolytic and raxolytic dehiscence. Kingdom Eumycota, true fungi, subkingdom Dicaria. In schizolytic succession, Fig 4.7c, the halves OPHA double septum at the base of the canidium split apart by the breakdown of a kind of middle lamella. In raxolytic succession, Fig 4.7d, the outer wall of a cell beneath or between canidia breaks down or ruptures. We will examine 10 different kinds of canidium development, 7 blastic, Fig 4.8, and 3 thallic, Fig 4.10. Type I, blastic acropetal candidiogenesis. The Manilia anamorph of Monalinia fructicola, the soft brown rot fungus off peach and other stone fruits, see Chapter 12, and the cladosporium anamorph of Mycos phaerella tassiana, an extremely common mold on decaying organic matter, and in the air, both produce canidia in chains by apical budding. The youngest canidium is at the tip of the chain. The chain branches when 2 buds, rather than 1, develop on the conidium. At the tip of the chain, this rather diagnostic conidium is then called a ramoconidium. This is clearly just a modified form of hyphal growth, Fig 4.8. Type 2, Blastic Synchronous Candidiogenesis. The Hyphamicites botrytis and Gonidobotrium produce many conidia synchronously on a swollen cell. Gonidobotrium goes on to form acropetal chains of secondary conidia by apical budding, while botrytis does not. Despite this deficiency, botrytis is one of the most successful molds, Fig 4.8. Other blastic synchronous molds are Chromolysporium and Botryosporium. Type IL, Blastic Sympodial Candidiogenesis. In species of Bovaria, insect pathogens which are now being used in biological control of potato beetle, the narrow apex of the candidiogenous cell extends sympodially. This means that each new apex becomes converted into a blastic conidium and then the next apex grows out from behind and to one side of it. The more conidia are produced, the longer the candidiogenous cell becomes. Although Leptographium anamorphs of Ophiostoma have single conidiophores, these have complex heads with several tiers of supporting cells, maculae, the ultimate ones in some species bearing many sympodially. Extending candidiogenous cells, and innumerable conidia accumulate in a slimy head. These spores are insect dispersed. Basofimbria, Teleomorph unknown, which is common on horse dung, has simple conidiophores that elongate sympodially during conidiation, Fig 4.8. Type 4, Blastic Analytic or Blastic Percurrent Candidiogenesis. In the Spilocea anamorph of Venturia iniqualis, the apple scab fungus, each seeding conidium leaves a ring-like scar, an annulation, around the candidiogenous cell, which then grows through the scar, percurrently, to produce the next conidium, Fig 4.8. Candidiogenous cells that have produced X spores bear X annular scars, hence the name analytic. Scopulariopsis, Fig 4.8, has several analytic candidiogenous cells on each branched conidiophore. Leptographium conidiophores may have many such cells at their apex, Fig 4.8. Chapter 4. A. B.C. Fig 4.9 Analysis of three modes of blastic candidiogenesis. Note that Leptographium has been mentioned under both sympodial and percurrent headings. It has recently been confirmed that some individual anamorphs can be both percurrent and sympodial. I'll give you two examples of how this knowledge may change our classification. When it was thought, not many years ago, that candidiogenesis in the synemedal anamorphs of Ophiostoma species was either exclusively sympodial or exclusively percurrent, they were segregated into two anamorph genera, Pesidium and Graphium. Now that it has been shown that both kinds of candidiogenesis can occur on the same conidiophore, they are being united again in the older genus, Graphium. In exactly the same way, some complex anamorphs of Ophiostoma species were segregated into the exclusively sympodial Verticillodiella, which I monographed, and the exclusively percurrent Leptographium, but are now united under the older name, Leptographium. Type V, Blastic Phylytic Candidiogenesis. Many common molds produce conidia in rapid succession from the open end of special candidiogenous cells called phylides. Common genera such as Penicillium, Aspergillus, Stachybotrys, and Cholera are all phylytic, Fig 4.8. Many plant pathogenic Hyphamicites, such as Fusarium and Verticillium, both causing serious wilt diseases of crop plants, also produce phylides. Penicillium and Aspergillus are dry-spored. Kingdom Eumycota, true fungi, subkingdom Dicaria. Fusarium, Verticillium, and Stachybotrys have slimy spores. Phylytic ontogeny is basically rather similar to type 4 percurrent, Fig 4.9a. Most phylides don't change in length while producing many successive conidia, although many wall layers build up inside the open end of the cell, Fig 4.9b. This accumulation of wall layers may eventually plug the opening, and in such phylides there is a tendency to produce sympodial extensions that develop new fertile apertures. Such phyalides are called polyphyalides since they have more than one candidiogenous locus. Type 6, Blastic retrogressive candidiogenesis. In Basopitospora, a thermotolerant fungus used in Indonesia in the preparation of a red food coloring, a canidium blows out at the tip of the initially undifferentiated candidiogenous hypha and is delimited by a basal cross wall, then a short zone of the hypha just below the canidium balloons out to produce the second canidium. After this has developed its basal septum, the next segment of the hypha just below plasticizes and blows out. This process can be repeated many times. As the chain of canidia elongates, the candidiogenous hypha becomes shorter. Similar development occurs in Trichothecium and Cladobotrium, but this is a very unusual kind of candidiogenesis in fact, prior to our time-lapse analysis of Basopitospora, it had been interpreted as being phyalitic, Fig 4.9c. Type 7, Basoxic Candidiogenesis. In the pseudoidium anamorph of Erisife gramineus, whitish or transparent bead-like chains of canidia, the powdery mildew, cover the host leaves. Each chain consists of a graded series of gradually maturing canidia, the oldest at the tip, the youngest barely differentiated from the hyphal cell just below it. New material is added at the base of the chain in a form of intercalary growth, arising from a sometimes swollen mother cell which appears to be a highly modified phyalide, Fig 4.21. Although powdery mildew anamorphs are almost ubiquitous, Basoxic candidiogenesis is not common among the anamorphic fungi. Type 8, Thalascarthric candidiogenesis. In the Geotrichum anamorphs of Dipodascus species, an assimilative hypha stops growing and then becomes divided up into short lengths by irregularly arising septa. These are double septa which split apart schizolytically to give a chain of short cylindrical fission arthrocanidia that disarticulates and appears jointed, hence arthric. In Oidiodendron, Fig 4.loa, a common soil mold, the branches of a tree-like canidiophore disarticulate into canidia, ultimately leaving only the denuded trunk, the stipe. Many basidiomycetes also produce thalascarthric canidia. Type 9, Thallic alternate arthric canidia. In Corymia, Fig 4.10b, some hyphal cells degenerate to release the intervening cells as raceolytically alternate arthrocanidia. This arrangement is also seen in Sporandoma, Amblyiosporium, and Geomyces but is not very common. Chapter 4. Type X, Thallic solitary candidiogenesis. The Microsporum anamorphs of Nannitsia, Fig 4.loc, which can digest keratin and cause skin diseases in humans, see Chapter 23, develop single, large, thallic phramospores at the ends of hyphae. These canidia are liberated raceolytically, as the final diagram shows. L. R. IL. J. I dash plus I. A. Oidiodendron thalascarthric. -1. 1. B. Corymia alternate arthric. C. Microsporum thallic solitary. Fig 4.10 Developmental analyses of Thallic candidiogenesis. Kingdom Mycota, true fungi, subkingdom Dicaria. One of the earliest kinds of developmental information was the observation that some molds produced canidia in chains, while others did not. But looking back over the various kinds of candidiogenesis I have just described, we find no fewer than 7 ways in which look-alike chains of canidia can develop. These are 7 good reasons we no longer rely on mature morphology alone to establish our classification of conidioid fungi. Importance of conidioid anamorphs. Why is it important to be able to identify conidioid fungi? Some of them literally grow on you. The most prevalent fungal diseases of humans, mycoses, are caused by conidioid fungi. Various superficial mycoses, ranging from ringworm of the scalp through jock itch to athlete's foot, are caused by keratin-attacking species of Microsporum, Epidermophyton and Trichophyton, see Chapter 23. When I compiled the fungi causing important plant diseases, I found that 62 were conidioid fungi, Hyphamicites and Chylomycetes, as compared to 111 from all other fungal groups combined. One of the most serious outbreaks of plant disease in the last half-century was the southern corn blight, caused by Drexlera, Helminthosporium, Medes, the anamorph of Cochliobolus heterostrophus, which devastated the U.S. corn crop in 1970. Unknown to farmers, the special Texas male sterile strain of corn that was becoming widely used for seed at that time was highly susceptible to the fungus, which produces a toxin that disrupts membranes, especially those of the mitochondria, reducing the production of ATP. The toxin also reduces photosynthesis. It inhibits uptake of potassium by the guard cells of the stomates, causing the stomates to close and thereby reducing the intake of carbon dioxide. Southern corn blight was brought under control by changing the strain of seed corn used by growers. Plant diseases are covered in more detail in Chapter 12. Cellulolytic Hyphamicitis cause blue stain and soft rot of wood and discoloration and loss of strength of cotton materials. The phylolytic Stachybotrys is particularly troublesome in the tropics, and can cause molding of almost any damp organic substrate. Stachybotrys chartarum is a common fungus growing on paper, such as that covering gypsum wallboard, in damp buildings and is now regarded in some quarters as a threat to human health, although there is little evidence to support this hypothesis. See Chapter 8. Many molds spoil food in storage, even in your refrigerator. Food spoilage is the subject of Chapter 20 and undoubtedly causes huge losses. These may be hard to quantify, since nobody writes it down when they throw out moldy food. Worse yet, Aspergillus flavus grows on and in peanuts and many other substrates, producing a mycotoxin called aflatoxin, which contaminates food and causes liver damage even at very low concentrations. It is perhaps the most potent carcinogenic substance known. Fusarium graminearum, growing on feed corn, produces another mycotoxin, zearalenone, which is a steroid and causes estrogenic syndrome, vaginal and rectal prolapse in young female pigs. Many other mycotoxins have been discovered in recent years. They are potential threats to human and animal health of which we are only now becoming fully aware, and they have necessitated the development of new. Chapter 4. Techniques for toxin monitoring and new programs for plant protection and food storage. The subject of mycotoxins is explored more fully in Chapter 21. On the positive side, Hyphamicitis and Chylomycetes anamorphs are among the prime colonizers and decomposers of plant debris, playing vital roles in the carbon and nitrogen cycles. Hyphamicites are important components of the saprobic soil fungi in many forests. The energy regime of many streams is based on the dead leaves of land-based plants. These are colonized by aquatic Hyphamicites, which usually form tetraradiate, four-armed, conidia, and are tolerant of low temperatures so can grow during the winter and even under ice. These fungi make the dead leaves much more palatable and nutritious for the various detritivorous invertebrates which eat them, and thus the fungi act as vital intermediaries in energy flow in northern stream systems. The terrestrial and aquatic ecology of conidioid fungi is explored in Chapter 11. Some soil-inhabiting Hyphamicites anamorphs have evolved specialized traps with which they catch small animals, nematodes, rotifers, tardigrades, amoebae, and even springtails. These truly predatory fungi can be visited in Chapter 15. In the same chapter you can read about fungi that make spores designed to adhere to, or be swallowed by, and infect tiny animals. Conidioid fungi are not just out there doing their own thing. We have also learned to exploit some of them for our own purposes. The enzymes of Penicillium camemberti produce the soft, smooth texture of camembert and brie cheeses. Penicillium roquefortii puts that inimitable zippy flavor in blue cheeses such as Roquefort, Danish Blue, Stilton, and Gorgonzola. Now there's even Cambozola, which blends the buttery texture of camembert with the assertive flavor of Gorgonzola. See www.mycology.com. Aspergillus oryzae is used in the Far East to turn soya protein into such delicacies as soy sauce, or its sweet Indonesian variant, ketchup, the word which became ketchup in English, see Chapter 19. Other anamorphs are also exploited in Asia to prepare a variety of fermented foods, and at least one, a species of Fusarium, is now mass cultured to produce food for people, Chapter 19. Finally, despite the insidious threat of mycotoxins, secondary metabolites of molds have come to play important roles on the human stage. For example, a substance which gave Penicillium chrysogenum an edge over competing bacteria in the natural habitat became one of our most potent weapons against bacterial disease, penicillin. Cyclosporin, a secondary metabolite isolated from the mold Talipoclidium nivoum, is one of the most effective and least toxic immunosuppressants yet discovered. It has enormously improved the success rate of organ transplant operations by preventing recipients' immune systems from rejecting the implant, but not leaving them totally defenseless against infection. This substance or its derivatives also hold out some hope for treatment of severe autoimmune diseases such as juvenile diabetes, rheumatoid arthritis, multiple sclerosis, myasthenia gravis, aplastic anemia, Addison's disease, Hashimoto's thyroiditis, and systemic lupus erythematosus, SLE. See Chapter 24 for a more detailed discussion of the cyclosporin phenomenon. In addition, conidial anamorphs can now be genetically transformed to act as hosts for vectors carrying multiple copies of genes from other eukaryotic organisms, including humans, and have already been persuaded to express and secrete a number of eukaryotic gene products, including insulin, human growth factor, human tissue plasminogen activator, used to dissolve blood clots, bovine chymosin, an enzyme used in Kingdom Eumycota, true fungi, subkingdom Dicaria, cheese making, and amylase and cellulase enzymes, see chapters 10, 24. Obviously, the biotechnological potential of the molds is tremendous. A survey of Askamycota holomorphs. Now to put Anamorph and Teleomorph together and talk about the whole fungus, the holomorph. I will briefly survey the more important orders of Askamycetes, linking the different life forms together in as many cases as possible. Although mycologists now recognize 60 orders of Askamycota, quite a few of them almost entirely lichenized, you will be, marginally, relieved to learn that I will mention only 22, mainly non-lichenized. Some of the larger lichenized orders are listed in Chapter 7. That is still a large number to deal with, and you will become familiar with them only if you see them and work with them. Phylum Ascomycota is now divided into 3 subphyla. Remember, you can Google any of these names, and Wikipedia will help you out: Taphragnomycotina, Saccharomycotina, and Pezizomycotina, the last being by far the largest, within which the orders fit as follows: Taphragnomycotina, 1, Taphrinoles, 2, Pneumocystodiales, 3, Schizosaccharomystales, Saccharomycotina, 4, Saccharomystales Pezizomycotina, 5, Pezizales, 6, Duthidiales, 7, Pleosporales, 8, Capnidiales, 9, Elaphomycetal, 10, Anigenales, 11, Urocials, 12, Labulbeniales, 13, Halatiales, 14, Rhitismetal, 15, Erysiphals, 16, Xyloreales, 17, Meleoles, 18, Microascales, 19, Hypocreales, 20, Sortareales, 21, Diaportales, 22, Ophiostomatal. Each order has its own visual and ecological flavor, which you can learn only by experiencing them. Many of them are easy to recognize after you have seen them once. Subphylum 1 Taphragnomycotina. Order 1 Taphrinoles, 9 genera, 120 species. This is an outlying group which causes serious diseases of some plants in the Rosaceae, example Taphrina defermans causing peach leaf curl, and the Salicaceae, Example Taphrina populina on poplar. Fig 4.11 shows Taphrina defermans attacking a peach leaf. Leaves become thickened, distorted, and often yellow or reddish in color. This fungus has 4 unique or unusual features. 1. The assimilative mycelium is dikaryotic. This would immediately distinguish it from most other Askamycetes. 2. It produces an exposed layer of acai on the surface of the host leaf. Fig 4.11. Since there is no surrounding or supporting fungal tissue, there is nothing we could call an ascoma. 3. The ascospores often bud in a rather yeast-like manner, even while still inside the ascus. 4. When the acai open to release their spores, they tend to split across the tip, rather than around it. Fig 4.11, so they are not like the rest of the operculate group. Compare them with the acai of the Pylobolus. Chapter 4. Hypertrophied peach leaf budding. Naked acai on peach leaf ascus. Ascospores budding dehiscence inside ascus host. Fig 4.11 Taphrinales, Taphrina defermans. Order Pneumocistidiales. These parasites are single-celled and amoeboid and closely associated with host lung cells. Thick-walled spherical cysts containing 8 spores eventually develop. The spores are released by rupture of the cyst wall. Meiosis may occur in the cysts, but we do not yet know the nuclear status of the different cell types, at least partly because none of the species will grow in pure culture. These primitive fungi have become notorious because one species, Pneumocystis jirovecii, causes a serious form of pneumonia very commonly found in AIDS patients and sometimes in other immunosuppressed or immunodeficient people such as those having had bone marrow transplants. Order 3 Schizosaccharomycetes, Schizosaccharomyces palm, also called fission yeast, has rod-shaped cells measuring 3 to 4x 7 to 14 microns. Cells grow at opposite ends and divide across the middle after laying down a transverse septum. In other words, they don't bud, but they do appear to be thallic and to have schizolytic succession. You can see how anamorphic terminology fits in. This species was originally isolated from millet beer in Africa. Palm is the Swahili word for beer, and its method of division has since made it a good experimental organism for yeast genetics and for studies of cell cycle. Nurse, Hartwell, and Hunt won the 2001 Nobel Prize in Physiology or Medicine for their work on these aspects of the fungus. The base sequence of its genome was published in 2002. It was only the 6th eukaryotic organism to have been entirely sequenced. Its genome has about 14 million base pairs, and it is estimated to contain just under 5,000 protein-coding genes. It has become an important model for studying cellular responses to DNA damage and the process of DNA replication. Kingdom Eumycota, true fungi, subkingdom Dicaria. As you can see from their position in the new phylogenetic classification, the Schizosaccharomycetes are not very closely related to most other yeasts. See Chapter 6. Subphylum 2 Saccharomycotina. Order 4 Saccharomycetes, the main group of yeasts. These, being rather different in appearance and behavior from most other fungi, are dealt with separately in Chapter 6. Check it out. For now I will content myself by saying that yeasts are frequently unicellular, although some produce hyphae, and are often involved in fermentative processes, such as the production of alcoholic beverages, bread, and some Asian fermented foods. Some yeasts are also active human pathogens. Yeasts usually exist in the anamorphic condition, see Chapter 6. Subphylum 3 Pezizomycotina. This huge assemblage embraces 10 major classes. I am going to list 6 of these, but only to establish baselines for the 22 orders I will briefly characterize. I will mention and illustrate examples of many of the families in the group because they are common, conspicuous, well-known, or particularly interesting. Most are illustrated at www.mycology.com. Class 1 Pezizomycetes. Order 5 Pylobolus have unitunicate operculate acai 150 genera, 900 species, the operculate Askamycetes. A. Family Pylobolus. Classic cup fungi producing apothecial Askamycota that are usually shaped more like saucers or goblets, usually without stalks, and found growing on wood, dung, or soil. They vary so much in size, color, texture, and ornamentation that most Askamycete specialists split the Pylobolus into several tribes or even families. Their asci have a diagnostic pop-open lid or operculum, Figs 4.3, 4.sa, and the tips of the asci are amyloid, sometimes expressed as I+ or J+. This means giving a blue, starch-like reaction in an iodine solution known as Melzer's reagent. A small species of Pylobolus often crops up on soil in greenhouses, frequently preceded or accompanied by its blastic synchronous Chromosporium anamorph, Fig 4.12a. Larger species of Pylobolus, producing thin, rather brittle apothecial Askamycota several centimeters across, with light brown or orange hymenia, can be found on the ground in spring and fall. The exposed hymenium of each of these Askamycota contains many millions of asci, and if you find a ripe specimen and blow on it, you can sometimes provoke a massive synchronized spore shooting, triggered by the change in humidity, in which the thousands of simultaneously expelled ascospores look like a puff of smoke. B. Family Sarcosomataceae. Wood-inhabiting fungi with apothecia that are often stalked, relatively tough, and brightly colored. The asci are suboperculate and non-amyloid. The scarlet cups of Sarcosiphycoxenia, growing from buried hemlock branches, brighten up the early spring in eastern Canadian woodlands. The brightly colored, stalked apothecia of Cucina are a common sight in the neotropics and may provide a camouflaged perch for the tiny but equally colorful poison arrow frog, as one National Geographic cover showed. Chapter 4. Blastic synchronous Canidia A, Chromosporium anamorph B, Pylobolus paraphyses operculum. Hymenium. C. Ascobolus. D. Scutellania. 1. Exterior E, helve/law. Interior. F. Tuber estivum. 2//// Tuber estivum tuber melanosporum. G. Asci and ascospores. Fig 4.12 representative Pylobolus, Unitunicati operculati, and Truffles. C. Family Otydiaceae. Scutellania scutellata, its orange apothecia rimmed with dark hairs, which give it the name eyelash fungus, Fig 4.12d, and producing non-amyloid asci, is one of the commonest cup fungi, growing on rotting wood. Anamorphs don't seem to be produced in this family. Some genera, such as Genea, Fig 4.13, produce closed but hollow Askamycota. The asci are cylindrical or clavate and are arranged in a flat hymenium lining the ascoma, but they do not shoot their spores. These apparently contradictory features show that members of this family are becoming sequestrate, meaning that their fruit bodies do not liberate spores at maturity, and hypogynous, which means that they produce. Kingdom Mycota, true fungi, subkingdom Dicaria. Pylobolus. Fig 4.13 suggested Evogenia. Like intermediary. 1 Eutionary sequence from Epigys to Hypogynous. Pylobolus. Geopora-like intermediary. Tuber. Their Askamycota underground. Another member of this family, Geopora, Fig 4.13, is also hypogynous, but now the air space inside the fruit body is much less than that in Genea, another step on the way to becoming a truffle, see Fig 4.12 and family Tuberaceae. This evolutionary process is diagrammatically illustrated in Fig 4.13. The fourth and final step sees the elimination of air spaces altogether and is a solid truffle of the genus Tuber, family Tuberaceae. Evolution toward the sequestrate and hypogynous condition is not restricted to the Otydiaceae but can also be seen operating in several other families of operculate Askamycetes. D. Family Ascobolaceae. Students who have followed the succession of fungal fructifications appearing on horse dung, and that should be all students of mycology, will be familiar with the two most important genera of this largely coprophilous family, Ascobolus and Saccobolus. Both produce minute, translucent apothecia, seen under the dissecting microscope, Figs 4.2, 4.4a, 4.12c. The dark dots are mature asci, which are broad and project from the hymenium when mature, so that their tips may orient themselves to point toward the light. The ascospores have a purple or brown outer wall layer. Ascobolus, Fig 4.2, like most other Askamycetes, shoots ascospores individually. Saccobolus atypically sticks all 8 ascospores together in a bundle, and they are. Chapter 4. Expelled as a single projectile, which gives them extra range, see the website. I haven't personally seen any Anamorphs in this family, although a few are known. Family Helvelaceae. These mostly spring-fruiting fungi have large and unusually configured apothecial ascomata. All are stalked, with beige to brown, hymenium-covered caps. Many Helvela species, Fig 4.12e, have a drooping flap on either side and for that reason are called saddle fungi. The ascomata of Gyromitra species are among the largest Askamycete fructifications. One species has the rather extravagant species epithet gigas, and some species contain the toxin gyromitrin, a precursor of the deadly monomethylhydrazine. By causing some fatal poisonings despite its name, the spring-fruiting Gyromitra esculenta, Fig 22.4, has earned its place in Chapter 22 on poisonous mushrooms. It is vital for morel hunters to be able to distinguish the convoluted head of Gyromitra, the false morel, from the ridged and pitted head of the delicious true morel, Fig 18.2, see below. F. Family Morchulaceae. While Gyromitra, above, is one of the few lethally toxic fungi, its cousin Morchula, the true morel, Fig 18.2, is one of the finest of all edible fungi. Species of Morchula have a broad, hollow stalk and a pitted and ridged, sponge-like, more or less conical head. Since the hymenium doesn't cover the ridges, it seems likely that a morel is a compound ascoma, each pit representing an individual apothecium. The anamorph of the morel is a blastic sympodial hyphamicete, Costantinella, which I often found growing on soil by trails in Algonquin Park, Ontario, in the fall. Note that the teleomorph fruits in spring. Morels have a broad geographic range but are common in relatively few areas, of which Michigan is perhaps the best known. People throng to the woods in May to hunt this elusive delicacy, and Boyne City hosts a popular annual morel hunting championship. When Dutch elm disease was killing millions of elm trees, morels sometimes fruited profusely around recently dead trees. In recent years they have also been collected in large numbers on burned-over areas of western forests. Morels are discussed as a delicacy in Chapter 18. Just to confuse the issue a little, a second genus of Morchulaceae, Verpa, also fruits in May. Species of Verpa aren't toxic, but neither are they good to eat. The wrinkled thimble cap, Verpa bohemica, looks like a morel, but it is easy to tell the difference by bisecting the fruit bodies. While the cap and stalk of the true morels are firmly united, the cap of Verpa is attached only at the apex. In addition, the stipe of Verpa is stuffed with cottony mycelium, while that of Morchula species is completely hollow. G. Family Tuberaceae, the truffles. Here, the evolutionary process still active in the Otydiaceae has run its course. The ascomata are sequestrate, hypogynous, and solid, no air spaces anymore, as you can see in the bisected specimen of tuber estivum in Fig 4.12f, which a truffle dog brought to me at Skegino in Italy. The asci of truffles, produced in a highly convoluted hymenium, are rounded and thin-walled, Fig 4.12g, with no trace of an operculum or other shooting mechanism, and usually contain only 1 to 3 spores. The ascospores of truffles have complex, highly ornamented walls. They come in two basic patterns, spiny and lacunose, Fig 4.12g. Only by examining a series of microscopic characteristics and by considering some intermediate forms that trace the probable course of evolution in the group, Fig 4.13, or by comparing DNA sequences can we tell that these fungi are related to the operculate Askamycota. Kingdom Eumycota, true fungi, subkingdom Dicaria. Although it doesn't make taxonomy any easier, we must now logically place these hypogeious, underground, families with their epigous, above ground, forebears in the order Pylobolus. The hypogeious habit has necessitated the evolution of new methods for passive spore dispersal, in which some agency other than the fungus supplies the energy for dispersal. Members of the Tuberaceae, especially species of the genus Tuber, the true truffles, have achieved this by developing what can only be called fascinating smells. These odors are released when the ascospores are mature and lead many mammals unerringly to the ascomata, which they unearth and consume, subsequently depositing the still viable spores elsewhere. Tuber melanosporum, the black diamond, queen, or Perigord truffle of French gastronomy, is dependent not only on mammalian vectors but also on the roots of oak and hazelnut trees, with which it establishes a symbiotic ectomycorrhizal relationship. See Chapter 17. Tuber melanosporum and tuber magnatum are, respectively, the black and white truffles of French and Italian haute cuisine, perhaps the most highly esteemed, and certainly the most expensive, of all edible fungi, and so are discussed in detail in Chapter 18. Class 2 Teleomorphs produce Bitunicate asci. 50 families, 650 genera, 6,300 species. Phragmoseptate or dictyoseptate ascospores are common in this class. In fact, if a fungus has ascospores of this kind, the odds are about 9 to 1 that it is a member of the Teleomorphs. Chylomycetes Anamorphs are common in this group. Order 6 Ideals. A. Family Venturiaceae. Venturia iniqualis causes apple scab, an economically important disease. You'll find the Spilocyapomi Hyphamicitis anamorph causing large brownish spots on the leaves and disfiguring blackish scabs on the fruit. It produces its blastocanelytic, percurrent, candidiogenous cells. In Fig 4.14a you can see the rings clearly, and obclavate canidia on those spots and scabs. But you won't find the teleomorph during the growing season. Its pseudothecial ascomata, seen in section in Fig 4.14a, develop slowly in the dead apple leaves over the winter, and the ascospores are shot in spring when the susceptible young leaves appear. Apiosporina morbosa causes the extremely common and disfiguring black knot disease of some rosaceous trees, especially wild cherry and damson plum. Its pseudothecial ascomata developing on conspicuous black fungal stromata. You shouldn't have too much trouble spotting these on wild cherry trees. This fungus damaged and disfigured my own damson plum tree despite my best efforts at controlling it by pruning. Order 7 Pleosporales. Pleospora occurs on dead herbaceous stems and has anamorphs in the common Hyphamicid genus Stemphylium, Fig 4.14b, and the Chylomycete genus Phama. The even more common mold alternaria is the anamorph, oft less frequently found Luia. Order 8 Capnidiales, commonly known as sooty molds. These fungi grow on the sugary excreta of various insects such as mealybugs and scale insects. I have found blankets of their thick black mycelia covering the trunks and leaves of southern beech trees, Nothofagus, in the forests of South Island, New Zealand. Fig 4.15a, b. The black, feathery branches of the numerous Anamorphs are easily seen by the naked eye, but soots frequently grow so intermingled that it is very difficult to sort out. Chapter 4. Spilocyapomi Anamorph. vs Pseudothecium. A. Venturia iniqualis. Osteole dictyoseptate ascospores RIILLN equals CLL // I_phaethi.rr / vs Pseudothecium dictyoseptate canidia. B. P/Eospora herbarum Ascostemphylum botryousum Anamorph Fig 4.14 Representative Duth ideals, Bitunicata. The various allegiances. Sooty molds occur in many warmer and wetter parts of the world. Class 3 Uroshiomycetes, many have prototunicate asci. Order 9 Elaphomycetal, 1 genus, 20 species. At first sight, the Hypogeious Ascomata of Elaphomyces look just like truffles, and they're even called deer truffles. But they have no discrete hymenium, though basically spherical. Kingdom Eumycota, true fungi, subkingdom Dicaria. Fig 4.15a, epiphytic sooty molds growing on a twig. B. Mixed colonies of sooty molds covering the normally light-colored bark of southern beech trees, not Hephagus, in New Zealand. A. B. Chapter 4. Non-shooting asci are produced randomly throughout the interior of the ascoma. Elaphomyces no longer offers much in the way of visual clues about its possible epigous ancestors, so only molecular techniques can help us decide its relationships. Order 10 Anigenales, 40 genera, 120 species. Here belong some unusual fungi which cause skin diseases in people and can digest hair, horn, see the website, and feather, all because some of them have the unusual ability to metabolize the resistant protein, keratin. Family Arthrodermataceae contains the infamous dermatophytes which cause superficial mycoses ranging from the inappropriately named ringworm of the scalp to another misnomer, athlete's foot. You certainly don't need to be athletic to catch it, see Chapter 23. Other members of the Anigenales can degrade cellulose, and yet others are coprophilous, dung inhabiting. They all produce ascomata, but although these are theoretically cleistothecial, their walls are often very loosely woven, and in some the ascospores can simply fall out through the gaps. The asci are always more or less spherical. Never shoot their spores, and tend to break down at maturity. Because of my earlier conclusion that asci evolved as spore-shooting devices, I assume that the ascoma and asci in the Anigenales are reduced forms, simplified during evolution from an earlier spore-shooting design. The ascomata often bear highly characteristic coiled or branched appendages that can make identification easy if the teleomorph is present. The ascospores. Appendages, ascomata, and anamorphs of various genera are illustrated in Fig 4.16. If you isolate dermatophytes in pure culture, they may or may not produce teleomorphs. But they will develop characteristic thallic conidile anamorphs, Fig 4.16. Sometimes these produce small, thallic-arthric conidia, chrysosporium or malbranchia. Sometimes large, spindle-shaped, transversely septate, solitary thallic conidia, trichophyton or microsporum, and sometimes the same culture will produce both kinds of conidia. When a fungus has two or more different anamorphs, these are called synanamorphs. The three most important anamorph genera of dermatophytes are Epidermophyton, Microsporum, and Trichophyton. Of these, Epidermophyton has no known teleomorph, 9 species of Microsporum have teleomorphs in Nannitzia, and 7 species of Trichophyton have teleomorphs in Arthroderma. Order 11 Urocials, 50 genera, 140 species. This largely cleistothecial order contains the teleomorphs of some of the most successful of all anamorphs, the common green and blue molds of the Hyphamicite genera. Penicillium and Aspergillus. Aspergillus conidiophores have characteristic apical vesicles, Figs 4.8 and 4.17a. The apices of Penicillium conidiophores are brush-like, Fig 4.8. These ubiquitous and almost omnivorous anamorphs are blastic phylytic and produce masses of dry, wind-dispersed conidia. These molds aren't just extremely successful, they are of considerable importance to us because they produce antibiotics and mycotoxins and cause huge amounts of food spoilage. Species of Aspergillus have teleomorphs in Eurotium. Sections through cleistothecial ascomata are shown in Figs 4.4d and 4.17a, or Emericella, while many Penicillia have teleomorphs in the rather similar Teluromyces or Eupenicillium. The cleistothecial ascomata of the teleomorphs have impermeable walls one or more cells thick. The asci, Fig 4.17a, are scattered throughout the cavity oft ascoma, i.e., never in a hymenium, they are spherical. Kingdom Eumycota, true fungi, subkingdom Dicaria. Families of the Anigenales. Peridia and substrate asterisk end. Family Ascospores appendages Anamorphs habitats. J5. Carnivore dung. Anigenaceae soil enriched with T. Keratin or dung. Pitted, spherical, chrysospore/um. F, and GT1. J, P. 1F /sporandoma keratin asterisk. Oblate, Al1 and to 1DF malbranchia. Boff. Arthrodermataceae decaying hoof, horn, feathers, hair, and skin. :::HOBL, low. J it /F. Some are oblate discold or chrysospore/um. Parastic on. Oblate convex -microsporum. Animals. Tr/Chophyton. Processed or decaying plant. Materials. Mixotrichaceae. Cellulose asterisk. Paper, straw, soil around the roots of plants. Decaying vegetation. Soil rich in organic matter. Gymnoscaceae. Variable asterisk. Smooth or slightly irregular, lumpy, often with polar. Various types of and/or equatorial dung. Thickenings. Fig 4.16 Families of the Anigenales. Chapter 4. And thin-walled and break down when the spores mature. The ascospores often have a pulley wheel shape. Again, it is thought that these fungi are reduced derivatives of spore-shooting ancestors. Slimy Canidia. Canidia ascospores dry __ and lt. / Peridium. Neck. Mononematous Canidia 4. Synemetal Canidiuma. Ascoma. I. Ascospores. Graphium Anamorph. Aspergillus Anamorph Teleomorph. A. Eurotium B. Ophiostoma U/Me. Fig 4.17 Prototunicata. A. Eurotials. B. Ophiostomatal. Class 4 Labulbeniomycetes. Order 12 Labulbenials, 75 genera, 1,700 species, Fig 4.18. All are invariably found attached to the exoskeleton of insects or occasionally millipedes and mites. The development of Stigmatomyces berrii, which is found on houseflies, is followed in Fig 4.18c. An ascospore becomes attached to the animal, germinates, and sends a foot into the exoskeleton to absorb nutrients. Although Haustoria may penetrate as far as the epidermal cells, puzzlingly there is never any real invasion of host tissues. The ascospore develops a median septum, and the upper cell becomes differentiated into a male organ, with several phyllide-like cells that produce spermatia. The lower cell then develops an ascogonium with a trichogyne, which is fertilized by the spermatia. Several asci then develop from the ascogonium and eventually deliquesce. The mature ascoma is spine-like, projecting from the exterior of the host, and can be seen with a hand lens. Other genera exhibit the same basic features, Corythromyces. Fig 4.18a, Hesperomyces, Fig 4.18b. The labulbenials apparently don't produce anamorphs, so are presumably spread from animal to animal by adhesive ascospores during mating of the hosts or when insects form dense swarms. This goes some way toward explaining the almost incredible site specificity of many labulbenials. Various species are restricted to one part of the insect, for example, one side of a particular left limb, or even to one sex of their host, although most species are not quite so limited. Kingdom Eumycota, true fungi, subkingdom Dicaria. A. Correthromyces. Age-germinated ascospore. Phyalides. B. Hesperomyces phyalides I. Young asci. C. Development of Stigmatomyces berrii. Fig 4.18 Representative label Biennials. A. Correthromyces. B. Hesperomyces. C. Stigmatomyces. Now for the classical unitunicate operculates. Although none have lids, opercula, the asci of this group are not as uniform in appearance or structure as we might like. Fig 4.19. Most have thickened walls at their tips pierced by a fine pore. Inside the apices, many have diagnostic sphincter-like rings, which control the expulsion of the spores. Some of those rings are amyloid, or J+, they stain blue in iodine, others don't react with iodine and are called chitinoid, some don't have rings at all. 88 Chapter 4 /T3 I /RL /1 0 11 190 10 I3 /I /0 I2 /II 1 in 11 2 2 / O I /I /1 I 11 /1 D 2 A B C D E F G. Fig 4.19 Unitunicate and operculate asci. A. Nectria, hypocreals. B. Sordaria, sordareals. C. Me/Anconese, diaportales. D. Claviceps, clavicipitales. E. Microglossum, amyloid ring, leodiales. F. Rose/Linea, amyloid ring, sphaereals. G. Lechinora, lechinorales. Class 5 Leotiomycetes, have unitunicate and operculate asci, order 13 heliteals, 13 families, 400 genera, 2000 species. These are the classical and operculate Discomycetes. The apothecial Ascomata are superficially similar to those of the Pylobolus but are often stalked. The asci are inoperculate and usually have amyloid apical rings. This suggests to me that the two major kinds of apothecial Ascomata are examples of parallel or convergent evolution. Several of the families in this order are common and well known, so 5 of them are dealt with below. A. Family Sclerotiniaceae. As the name implies, these frequently parasitic fungi often form sclerotia, which may be solid masses of fungal tissue or may be of mixed origin fungal hyphae riddling a mummified host such as a peach, plum, or cherry or a tree catkin. Having overwintered in this guise, they germinate in spring and use the stored energy to produce stalked apothecial Ascomata. Fig 4.20a. Ascospores, the primary inoculum, are shot when the host is in flower and gain entrance through the stigma. The Anamorphs are generally responsible for secondary dispersal during the growing season, and some cause serious plant diseases. For example, the soft brown rot of peaches, Fig 4.20a, and cherries is produced by a Manila anamorph of Monilinia. The beige or grayish powder on the surface of the peach and the cherry are made up of branched chains of Canidia. The longer I left the ripe cherries on my cherry tree, the more of them succumb to the Monilinia soft brown rot, as the Canidia being produced on one cherry infected others. See Chapter 12. Another Monilinia produces spur blight of wild cherry, killing back young shoots and forming new Canidia on the leaves. Many members of this family have distinctive anamorphs, while the teleomorphs are relatively uniform. So some of the genera erected for the teleomorphs have rather atypically been distinguished by characteristics of their anamorphs and even named after them. So we have Sclerotinia with Sclerotium, Sclerotial, Anamorphs, Monilinia with Manilia Anamorphs, Blastic Acropital, Botrytinia with Botrytis Anamorphs, Blastic Synchronous, and Streptotinia with Streptobotrytis Anamorphs, Blastic Sympodial. Sclerotium, Manilia, and Botrytis cause serious plant diseases. Gray mold of strawberry is caused by Botrytis cinerea, Chapter 12, but when Botrytis grows on overripe grapes in certain areas of Canada, France, Germany, Hungary, and South Africa it is. Kingdom Eumycota, true fungi, subkingdom Dicaria. Manilia Anamorph stalked Apothecia paraphysis sporulating on peach mummy. Blastica cropetal. _. Canidia. I/CJ. ID. IO. U. O. 0. 0. A. Monilinia. Ascus. Tongue-shaped Apothecia B. Geoglossum ascus 0. If Cascaspores 7L1 / I comma : ti host and fungal roof I stalked Apothecia appendaged canid 1 /Phyolide /JF1 // I /ITTI *IV. Espignidia. C. Leatia. Fruiting on Vs Apothecium Certhospora Anamorph. Conifer needles. D. Facidium. Fig 4.20 representative in Operculate Askamycota. A. Monilinia and its Manilia Anamorph. B. Geographic / awesome. C. Leatia. D. Facidium and its Certhospora Chylomycetus Anamorph. Called the noble rot, Puriture noble, Adelphii ul, because the small quantities of sweet dessert wine that can be made from such shriveled grapes have intense and exquisite flavor and can be sold for very high prices. Find out what a bottle of Chateau d'Yquem Sauternes from France, or a Trockenbeerenauslese from Germany, costs at your local wine store. Be prepared for a shock. The full story can be found in Chapter 19. Chapter 4. B. Family Facidiaceae. Some Facidium species cause snow blight diseases of conifers. If we look more closely, we will see that this family is not typical of Askamycota fungi in general, since the Ascomycota develop inside host tissue and are at first covered by a roof of dark fungal tissue, Fig 4.20d. But at maturity the roof splits open and exposes the hymenium. The apical ring in the asci is amyloid, I+. Compare this family with order 14, Rhytismetal, below. How do the orders differ? Facidium has Chylomycetus anamorphs, those of pathogenic species such as P. coniferarum belong to Apostracerea, while those of saprobic species like P. bitulinum belong to Certhospora, Fig 4.20d. C. Family Geoglossacea. Literally earth tongues produce unusual stalked, somewhat flattened and tongue-like ascomata which emerge from the ground, Fig 4.20b. The hymenium doesn't line a cup or saucer but covers the convex upper surface of the ascoma, which is fleshy and yellow in Spathularia, tough and black in Microglossum, Geoglossum, and Trichoglossum. If you squash a tiny piece of the hymenium of a mature Geoglossum ascoma, you will see the asci, each of which contains a bundle of 8 long, parallel, phragmoseptate brown ascospores, Fig 4.20b. D. Family Leatiaceae contains more typical Askamycetes, such as Bisporella, which produces those tiny bright yellow discoid apothecia so common on fallen, decorticated tree trunks, while Chlorociboria, also fairly common, stains wood green and forms small green apothecia. Less typical are the spectacular ascomata of Leatia, Fig 4.20c. These are much larger, are stalked and jelly-like, and have convex fertile heads, a beautiful velvety green in Leatia viscosa, that contrasts with its vivid yellow stipe. Another rather spectacular member of this order is Bulgaria inquinans, found on wood of deciduous trees. The clustered apothecial ascomata have a rubbery texture, and the hymenium is jet black. All of the above are illustrated on the website. E. Family Dermatacea includes Diplocarpon rossi, which, with its Marsanina Chylomycetus anamorph, causes the dreaded black spot of roses, and another common fungus, Trochala ilicicola, which fruits on the upper surface of dead leaves of holly, ilex, in our garden. Its ascomata have a hinged lid which opens when the leaf is kept in a damp chamber. Order 14 Rhytismetal, 70 genera, 400 species. The Ascomycota, like those of the Phacidiaceae, develop immersed in host tissue or a fungal stroma, which ultimately ruptures to expose the hymenium. The Asci often have apical rings, but these are small and chitinoid, do not stain blue in iodine. The ascospores are usually long and thin and have a slimy sheath, absent in the Phacidiaceae. The genus Lophidermium is sometimes endophytic and asymptomatic in pine needles for much of its life but eventually fruits after the needles die, see Chapter 11. Rhytisma acerinum causes tar spot of red maple leaves in eastern North America. Rhytisma punctatum produces a similar syndrome on big leaf maple in western North America, but the small, individual Ascomycota do not fuse, see the website. Order 15 Erysiphals, 28 genera, 100 species. All members of this order are obligate parasites on leaves and fruits of higher plants, causing diseases called powdery mildews. These fungi have a superficial mycelium which extracts nourishment from the host plant through specialized hyphae that penetrate the epidermal cells of the host and develop absorbing organs called haustoria, Fig 4.21. You should have no difficulty spotting a few powdery mildews in summer because their whitish colonies growing on living leaves are unlike anything else. In dry kingdom Eumycota, true fungi, subkingdom Dicaria. Ascometal appendages Phylactinia, and gt, _____. See Erysife Microsphaera digitate haustoria. Fig 4.21 Representative erysiphals. Summers they are particularly common on grass in shady parts of lawns, on squash plants, cucurbits, on perennial phlox, on Alnus rugosa, and on many other angiosperms. Basoxic chains of Canidia of the oidium like Anamorph, Fig 4.21, whose powdery, whitish appearance gives these diseases their name, arise from the mycelium in early summer. Airborne Canidia spread the disease from plant to plant and are later succeeded by dark Ascomycota, which mature slowly in fall and release ascospores the following spring. This order parasitizes well over 1,000 higher plant species, and the powdery mildews of grapes, hops, gooseberries, and cereals are economically important. Diseases. Chapter 4. The generic concepts in the teleomorphs of this order are unusually straightforward and easy to apply, since they depend on two major features of the ascoma: 1. The number of asci within it, and 2. The kind of appendage growing out from it, Fig 4.21. In one way, the erysiphals are the antithesis of the Sclerotiniaceae. There, the Anamorphs were far more distinctive and diverse than the Teleomorphs. Here, the reverse is true. Most Anamorphs of the erysiphals belong to the Hyphamicite genus Oidium or other very similar genera. Although the order Erysiphals is very easy to characterize and recognize, its systematic position is controversial. Some mycologists insist that its asci are bitunicate, which would place it alongside the Duthideals, see above, but many mycologists do not accept this and place the order among the unitunicate Askamycetes. The asci are sometimes rather thick-walled, but one of the world experts on the group, Dr. Zheng Ruyang of Beijing, told me that she had seen distinctive inner and outer wall layers only in an undescribed taxon from Tibet and had never seen the jack in the box mechanism so typical of bitunicate asci. The asci seem to have neither an operculum nor an apical ring apparatus. This information, plus their strange arrangements for dispersal and dehiscence, see Chapter 8, their unique basoxic anamorphs, and their obligately parasitic yet strangely superficial lifestyle, make them a rather atypical, although important and interesting, group. Alder, Alnus, leaves often bear extensive colonies of Phylactinia. Under the dissecting microscope you can see Ascomycota with unique appendages, Fig 4.21. The basal bulbs of the appendages develop first, then the needle-like extensions grow out. At maturity, these appendages all bend downward in unison and lever the ascoma off the leaf surface, breaking its connections with the mycelium. It is then free to be blown or splashed away, becoming attached to a new substrate by an apical blob of mucilage secreted by specialized hyphae. Paradoxically, this leaves the asci, which are designed to shoot their spores, facing downward. The final chapter is written when the ascoma splits around the equator at a built-in line of weakness and hinges open so that the spores can finally be shot away. Fig 8.3. Class 6 Sordarioomycetes have unitunicate and operculate asci, order 16 xyloreals, 225 genera, 1,300 species. Many members of this order produce dark, brittle, globose to pear-shaped individual parathecial ascomata with prominent osteoles, narrow apical openings, Fig 4.22a. Others have many parathecial cavities immersed in a single stroma to form a compound. Fructification, Fig 4.22c, d. The asci often have an apical ring or sphincter, which is usually, although not always, amyloid, stains blue in iodine. Thread-like, sterile elements called paraphyses are present between the asci in the hymenium of some members but absent from others. Ascospores can be light or dark, simple or septate, with or without germ pore or slit, sometimes with gelatinous sheaths or appendages. The compound fructification of Xylorea, a common wood-inhabiting genus, Fig 4.22c, has hundreds of individual parathecial cavities just below the surface. Each of these contains many asci. The asci are inoperculate, with an amyloid apical ring, and contain 8 darkly pigmented, asymmetrical ascospores. These will eventually be shot out through the narrow osteole. Kingdom Eumycota, true fungi, subkingdom Dicaria. A. Podospora 8, Chaetomium, Sordarioles. Ascus ascospore ascus. Sordarioles. I. Vs compound //. C. Ascoma. D. Diatropy, Diatropoles. Fig 4.22 representative parathecial Askamycetes. This order also includes such pathogens as Hypoxylon mammatum, which causes poplar canker, a disease that kills millions of trees every year. The extensive, more or less elliptical cankers develop groups of parathecial ascomata after the tree cambium has been killed. Family Diatropaceae, with its extensive stromatic multiascomatal fructifications spreading out under bark off trees, and its tiny sausage-shaped ascospores, Fig 4.22d, has now been folded into the xyloreals. I. 1. Compound ascomata on branch. T.S. sterna. Amyloid apical ring R. Apical ring 7 / F /I1I. I. Ascus. I. I. 1. T.I. H. I. Chapter 4. Order 17 Meleoles. These are in some ways tropical, darkly pigmented analogues of the erysipelas, powdery mildews. They are obligate biotrophs that live superficially on their hosts, anchored by numerous conspicuous hyphopodia and deriving food from haustoria that penetrate only the epidermis. Although their spreading black colonies look ominous, they do not usually cause serious damage to their hosts. See the website. Order 18 Microascales. Ceratocystis fagacerum and its Caloricaerchina phyolitic anamorph are the cause of another widespread and serious tree disease, oak wilt. The teleomorphs of Ophiostoma, Ophiostoma tal, below, and Ceratocystis are very similar, but the genera are easily distinguished by their anamorphs. The Calora anamorphs of Ceratocystis have solitary phyalides with long, tubular collarets and form long, cylindrical conidia, Fig 4.8. Order 19 Hypocreales, 80 genera, 550 species. This order is recognized by its brightly colored, simple or compound, parathecial ascomata usually yellow, orange, or red, which are fleshy or waxy in texture and usually borne on a supporting layer of mycelium, a subiculum, or in stromata. 4 genera are especially well known, and the family Clavicipitaceae is among the most fascinating of all fungi, as you will read. A. Nectria, Fig 4.23, often has bright red, superficial perithecia containing toothed Didymosporous, ascospores. Some species cause cankers and diebacks of trees. Nectria sensulato has a variety of conidile anamorphs, Fig 4.24, but all of them are phylytic. The erumpent sporodicia of one commonly encountered anamorph, tubercularia. Fig 4.23, cause a condition known as coral spot. As you can see, the tubercularia anamorph often grows beside the dark red Nectria perithecial Ascomycota. It is interesting and a little unusual to see both phenotypic expressions of the genome being produced simultaneously. However, the most economically important of the nectriaceous anamorphs are certain Fusarium species, Figs 4.24, 14.2, 21.lb, many of which cause destructive wilt diseases off higher plants or produce dangerous mycotoxins. B. Gibberella also has Fusarium anamorphs. A member of this genus causes a disease of rice called foolish seedling, in which seedlings grow too rapidly and consequently fall over. The active principle, a plant growth hormone called gibberellic acid, has been extracted and is now widely used to study and stimulate plant growth. C. Hypomix lactifluorum is an orange-red fungus which parasitizes the agaric mushroom, Generolactorius and Russula, producing a layer of tissue that completely covers the gills and suppresses their development, and then develops thousands of bright orange-red perithecial ascomata all over the surface of the host. The hypomix completely envelopes the aborted mushroom, and its color gives the host parasite combination the name lobster fungus. Strangely enough, this monstrosity is edible, although I regret to have to tell you that it does not taste like the divine crustacean. Hypomix has extremely characteristic spindle-shaped, toothed, colorless ascospores. The anamorphs of Hypomix species belong to the Hyphamicid genus Cladobotrium, which has an unusual blastic retrogressive method of forming canidia, type 6 above. D. Hypocrea forms fleshy stromata on wood. The dark spots on these stromata are the osteoles of the embedded perithecial cavities. The teleomorph of Hypocrea. Kingdom Eumycota, true fungi, subkingdom Dicaria. Teleomorph anamorph. Ascomata and Canidiomata on branch. Vs perithecial stroma. Perithecial cavity. Host. End LT. C, Canidiophores. Single. Coidia tubercus/aria. TL1111 TT art canidiophore. With phylides. Host. Vs sporodiceal canidioma of tubercularia anamorph. Fig 4.23 Hypocreals, Nectria cinnabarina and its tubercularia anamorph, although relatively common, is recorded far less often than its green-spored, phylytic anamorph, Trichoderma, Fig 14.3, which, because it is a broad-spectrum mycoparasite and produces cellulases and antibiotics, is one of the most important molds in forest soils. It is now being exploited in biological control of pathogenic fungi, see Chapter 14, and in the production of enzymes which can convert cellulose to glucose, Chapter 24. Trademark T. Chapter 4. ZY1L-10 stroma. S. Rhomatographium. FF90 OOIO. Cutilakes.psis/STLLBELLAVOLUTELLA. Dendro MYRO. O. Dash. Dot I. A dot comma. Dash. Verticillium. 00. B. Backslash. A dot B. Lindrocarpon. Fusarium, A. Glyoclad. Rose /JMG catenulatum. Acremonium. Fig 4.24. Hypocreals, Anamorphs of Nectria sensulato. Arrows indicate possible lines of derivation. Family Clavicipitaceae, 27 genera, 270 species. This order comprises a group of highly evolved and sophisticated, all obligately parasitic fungi with a. frequently stalked, all fungus stromata, Fig 4.25b, b. very long asci without apical rings but with thickened tips, Fig 4.25f, and c. extremely long, thread-like ascospores that in some taxa fragment at or following release, Fig 4.25f. Three bizarre and spectacular genera, Claviceps, Cordyceps, and Epiclo, will give us a snapshot of this fascinating family. A. Claviceps purpurea, Fig 4.25 ac, discharges its ascospores when its main host, rye, is in flower, and infection takes place through the stigma. As the infection progresses, the fungus takes over the food being channeled into seed production by the host. The ovarian tissues are replaced by a mycelial mat that produces masses of canidia. Offs facilia anamorph in a sweet-smelling nectar. Insects are attracted to the nectar and spread the canidia to other host plants. The mycelial mat hardens and becomes a purplish sclerotium, the ergot which replaces the grain, Figs 4.25a, 4.26. I have found very large ergots on Elymus mollis, a large grass that grows along the shore in the Pacific Northwest. The largest ergot was 4 centimeters long and almost 5 millimeters wide. These sclerotia fall to the ground in autumn, overwinter, and germinate the following spring, each producing several stalk stromata, Fig 4.25b, c. Each stroma has a spherical head within which many parathecia develop just below the surface. Each parathecial cavity contains many asci, each ascus having 8 extremely long ascospores that are forcibly ejected at maturity. Because this fungus has a small target, the stigma of the grass flower, which is available only during a narrow time window, and because spores reach it only by chance, the fungus must disperse a large number of ascospores in a short time. A rough compound Ascomycota /I2I /ergot /2I/1IL/1/T//Kingdom Mycota, true fungate subkingdom Dickeriae/III/C, vs compound Ascoma, stroma, compound Ascomycota osteoles ipart.comma-spores Ascus elaphomyces, host, 97 compound Ascomycota /I2I /ergot /2I/1IL/1/T//Kingdom Mycota, true fungate subkingdom Dickeriae/III/C, vs compound Ascoma, stroma, compound Ascomycota osteoles ipart.comma-spores Ascus elaphomyces, host, 97 /I, comma / Fig 4.25 representative Clavicipitaceae. AC, Claviceps, DF, Cordyceps. Calculation suggests that a single ergot can give rise to 5 stromata, and each of those may contain 100 parathecial cavities, each cavity with 50 asci, and each ascus producing 8 ascospores, a total of 5 x 100. x 50 x 8 equals 200,000 propagules per ergot. If the sclerotia are accidentally consumed by cattle, or if rye bread made from ergoty rye is eaten by humans, a large number of alkaloids found in the ergot cause a form of poisoning known as ergotism, or, more picturesquely, St. Anthony's fire. Human victims frequently hallucinate and feel that they are burning. See Chapter 21 for a fuller account of this mycotoxiciosis. The alkaloids ergotamine and ergotyline. Chapter 4. Fig 4.26 Ergots on a grass cause contractions of the smooth muscles, and the ensuing restriction of the peripheral blood supply can lead to gangrene and even death. St. Anthony's fire was fairly common in the Middle Ages, and sporadic outbreaks occurred until recently. Ergot, the only fungal structure in the British Pharmaceutical Codex, has been used in obstetrics, both to induce childbirth and to control postpartum bleeding. Another species of claviceps brought the genus renewed fame, or perhaps I should say notoriety, as the prime source of LSD, lysergic acid diethylamide, one of the most powerful psychedelic drugs. It is 100 times more potent than psilocybin, the active ingredient of magic mushrooms. B. Cordyceps species, Fig 4.25 df, are bizarre. They generally parasitize insects or spiders or hypogeious, underground, fungi, and their large stromata spring up directly from their victims. These parathecial stromata arising from an insect larva or pupa are known as vegetable caterpillars, in recognition of the fact that they always incorporate elements from more than one kingdom. These strange two-kingdom structures, see the website, are used in traditional Chinese medicine as a treatment for many conditions. General debility after illness, weakness, spitting of blood caused by TB, chronic coughing and asthma, night sweating, anemia, and even malignant tumors, you name it. As mentioned, a few species of cordyceps don't pick on arthropods but cannibalistically attack another fungus. Actually, it's even another Askamycota underground. Lnltingdomycota, true fungi, subkingdom Dicaria. Deer truffle, Elaphomyces. The large, stalk stroma of the cordyceps can be seen emerging from the host truffle in Fig 4.25e. Every September for many years, during a mycology field course I taught, we found cordyceps parasitizing Elaphomyces along one of the hiking trails in Algonquin Park, Ontario. Once one of the students had spotted the dub-shaped stroma of the parasite. Excitement ensued as we dug down, following the yellow rhizomorphs of the fungus, until we finally excavated the host. This find was often dubbed, and with good reason, fungus of the day, although perhaps the first word of that title should have been pluralized. Cordyceps, which must infect target organisms that are clearly far scarcer than rye flowers, goes a big step further than claviceps in the multiplication of spores. Each of the 8 long ascospores breaks up into about 100 part spores, often while still in the ascus, Fig 4.25f. I estimate that the usually single large stroma produced by this genus from its fungal or insect host may bear as many as 800 parathecial ascomata, each containing at least 100 asci, each ascus containing 8 spores, and each of them fragmenting into 100 part spores, for a total of 800 x 100 x 8 x 100 equals 64 million propagules. 64 million airborne spores from a single stroma. In 1996, Cordyceps subsessilis was discovered to be the teleomorph of Talipocladium nivoum. So what? You might say, until you realized that Talipocladium injalatum is the fungus that produces the medically important, selective immunosuppressant cyclosporin, which has made the organ transplant revolution possible. For the story of that amazing pharmaceutical, see Chapter 24. C. Apical causes choke disease of grasses. A grass called Glyceria normally produces open, nodding inflorescences. When Apical attacks, the energy for the inflorescences is stolen by the fungus and used to produce creamy yellow parathecial stromata, each incorporating many parathecia, which surround the stalk of the now sterile grass. See the website. In a recently discovered twist to this story, this apparently damaging parasitic fungus has been found to have a mutualistic symbiosis with the grass. Epiclose simple, phyolitic anamorph, neotyphidium, which looks very like a cremonium, grows systemically throughout the grass plant without producing any disease symptoms and actually protects the grass from herbivores by producing a virulent neurotoxin. A more detailed discussion of this relationship is given in Chapter 21. Order 20 sort Aerials, 5 families, 75 genera, 600 species. This is a generally saprobic group producing solitary parathecial ascomata and found on dung or decaying plant remains. Their asci sometimes have non-amyloid apical sphincters and sometimes lack any apical apparatus. Several members of this order are important tools in fungal genetics and biochemistry. First and foremost is Neurospora, which has justifiably been called the Drosophila of the fungus world. It was on Neurospora crassa that the science of haploid genetics was founded. The uses of Neurospora and Sortaria mutants are explored in Chapter 10. Many species of Sortaria and Podospora. Fig 4.22a, fruit on herbivore dung and shoot their ascospores from parathecial Ascomata whose necks are phototropic, point toward the light. Different members of genus Podospora, which has over 100 species, have 4, 8, 16, 32, 64, 128, 256, 512, 1024, or 2048 ascospores per ascus, see the website. The various combinations of tubular and gelatinous ascospore appendages in Podospora not only help in species identification but also stick the spores to grass after they have been shot away from the dung on which the ascomata develop. Some species of Podospora have Phyllophora. 100 Chapter 4. Anamorphs, Fig 4.22a. Chaetomium, Fig 4.22b, is an important cellulolytic genus that damages fabrics and paper, especially in the tropics. It is unlike most other Sortarials in that its asci, although cylindrical, deliquesce or autolyze at maturity. Since they don't shoot their spores, they have no apical ring mechanism, and the mucilaginous, lemon-shaped, or American, football-shaped ascospores ooze out of the ascoma into a characteristic mass of coiled or dichotomously branched hairs that develop on the top of the ascoma. Dispersal must be by rain or arthropods. Chaetomium has Botryotrichum anamorphs, Fig 4.22b. Neurospora has Chrysanilia anamorphs, which look very like Manilia, although that genus belongs in order 13, the heliteals of class Teleomycites. Order 21 Diaport Hales, 90 genera, 500 species. Here several beaked, parathecioid ascomata are usually immersed in a single stroma, as in Diaporthi impulsa, Fig 4.27. The asci, which have an amyloid apical ring, become free inside the ascoma and then autolyze. This rather paradoxical situation suggests that evolution is in active progress here. Two important genera stand out. Cryphonectria. Endothea, parasitica causes chestnut blight, which almost extinguished an important species of North American tree in about 50 years, you can read the full story in Chapter 12. Because of this near extinction, you will probably not be able to find specimens of Cryphonectria, but another member of this order, Geomonomyces graminans, which causes whiteheads or take all of wheat, is common. It rots the roots of afflicted plants and so causes premature drying out of the plant, sometimes reducing yields to zero. Immersed parathecia. Fig 4.27 Diaport Hales, Diaporthi impulsa. Osteoles. Host periderm. Order 22 Ophiostomatal, 15 genera, 130 species. The ascomata of this order generally have long, tubular necks, with the osteole at the tip, Fig 4.17b. The asci are not arranged in a hymenium and autolyze early. The spores ooze out of the osteole and form a slimy droplet that is supported by a radiating ring of Kingdom Eumycota, true fungi, subkingdom Dicaria. Specialized, hair-like hyphae at the top of the neck. These fungi often fruit in bark beetle tunnels, and the elevated spore drop has evolved to ensure that the beetle carries spores with it when it flies off in search of another tree. The most important genus in this order is Ophiostoma. Ophiostoma ulmi, Fig 4.17b, causes Dutch elm disease, which was introduced into the United States in 1930 and into Canada in 1944, has since spread right across the continent, and has much more than decimated the American elm. To decimate means to take out 1 in 10. This beetle-transmitted fungus has a graphium anamorph that produces many tall, cinematal Canidiomata, Fig 4.17b, each bearing a slimy droplet of Canidia at its tip. In producing this stalk spore drop, the anamorph is completely analogous to the teleomorph. Both have evolved to ensure that beetles don't leave home without them on their way to the next tree. It has proved very difficult to breed resistant strains of the American elm, Ulmus americana, but some naturally resistant trees have been found, and scientists are now multiplying these in cell culture with a view to generating millions of resistant trees that can replace the magnificent vase-shaped elms that not long ago shaded many city streets in North America. See the website. Mostly, dichotomous key to some groups of Askamycetes. 1. No ascoma produced, asci solitary, in chains or in layer on host. 2. 1 ascoma produced, open or closed, epigys above ground or hypogynous below ground. 3. 2 hyphae often absent, ascus like myosporangia free or produced on individual hyphae or absent. Yeasts, Chapter 6 Saccharomycetes, Schizosaccharomycetes. 2 hyphae always present, asci born in a naked layer on the surface of the host plant. Taphrinoles. 3 thallus containing algal cells or filaments. Lichens, Chapter 7 Lecanoromycetes, Chaetotheriomycetidae. 3 thallus without algae or cyanobacteria. 4. 4 ascus wall thick, with 2 functionally different layers, bitunicate. Duthidiomycetes, Duthidiales, Pleosporales, Botryosphaereals, Capnidiales. 4 ascus wall thin, functionally single-layered, lissing at or before maturity. In some orders. 5. 5 ascus wall lissing before spore maturity, ascospores not discharged. Prototunicate. 6. 102 Chapter 4. 5 ascus wall persistent, ascospores discharged except in hypogynous forms, unitunicate. 9. 6 assimilative hyphae absent, ascomata on exterior of insects, spine-like outgrowths. Labulbeniomycetes, labulbenials. 6 assimilative hyphae well developed, immersed, or superficial. 7. 7 ascomata usually with ostiole, asci lissing early. Ophiostomatal. 7 ascomata closed, cleistothecial, occasionally osteolate, asci spherical, randomly arranged. 8. 8 ascomatal peridium complete, ascus wall lissing before maturity, anamorphs usually blastic phyolitic. Urocials. 8 peridium often loosely woven, with appendages, anamorphs thallic. Anegenales. 9 asci operculate, or with thin apex, ascomata apothecial or hypogynous. 10. 9 asci opening by apical pore or canal, with apical sphincter or thick apex, inoperculate, ascomata parathecial or apothecial. 11. 9 asci without lid or sphincter, ascomata closed, obligate plant parasites with superficial assimilative hyphae. Erysiphals. 10 ascomata apothecial or hypogynous, asci in hymenium. Pezizales. 10 ascomata closed, hypogynous, asci random. Elaphomycetal. 11 mature ascomata have an exposed hymenium. 12. 11 mature ascomata parathecial, closed but with ostiole. 13. 12 asci with apical sphincter bluing in iodine, amyloid. Leotiales. 12 asci with non-amyloid sphincter. Rhitismatal. 13 asci with apical sphincter amyloid, bluing in iodine. 14. Kingdom Eumycota, true fungi, subkingdom Dicaria. 13 asci with non-amyloid ring or ring absent. 0.15. 14 ascomata compound, parathecia embedded in a black stroma, ascospores small, sausage shaped, allantoid. Diatripoles. 14 ascomata single, or compound in a stroma, ascospores not sausage shaped. Xyloreals. 15 ascomata single. Sword areals. 15. Ascomycota grouped in a stroma or on a subicular layer. 16. 16. Stroma often stalked, asci long, narrow, lacking sphincter, but apex thick, with pore, ascospores. Long and thread-like, often fragmenting at maturity, all obligately parasitic, on plants, arthropods, or fungi. Clavicipitaceae 16. Stroma never stalked, asci and ascospores not as above. 17. 17. Ascomycota compound, parathecia immersed, with long neck or beak, asci with apical ring but lissing. Diaport hales. 17. Parathecia not beaked, often brightly colored, embedded in a stroma or superficial on a subiculum, asci not lissing. Hypocreals. Further reading on Askamycetes and their Anamorphs. Arx, J. A. von. 1981. The genera of fungi sporulating in pure culture. Vedas, Germany, Kramer. Arx, J. A. von, and E. Muller. 1975. A re-evaluation of the Botanicat Askamycetes with keys to families and genera. Studies in Mycology 9. Barn, Netherlands, Centraal Bureau voor Schimmelcultures. Barron, G. L. 1968. The genera of Hyphamicites from soil. Baltimore M.D., Williams and Wilkins. Breitenbach, J. and F. Kranzlin. 1984. Fungi of Switzerland. Volume 1, Askamycetes. Lucerne, Switzerland. Verlag Mycology, the gold standard for books on Askamycetes. Carmichael, J. W., B. Kendrick, I. L. Connors, and L. Sigler. 1980. Genera of Hyphamicites. Edmonton, Canada, University of Alberta Press. Cole, G. T., and B. Kendrick, eds. 1981. Biology of Conidioid Fungi. 2 volumes. New York, Academic Press. Cole, G. T., and R. A. Sampson. 1979. Patterns of Development in Conidioid Fungi. London, Pitman. Dennis, R. W. G. 1968. British Askamycetes. 2nd ed. Lehr, Germany, Kramer. Domsch, K. H. W. Garns, and T. H. Anderson. 1980. Compendium of Soil Fungi. Volumes 1 and 2. London, Academic Press. 104. Chapter 4. Ellis, M. B. 1971. Dematiaceous Hyphamicites. Kew, UK, Commonwealth Mycological Institute. 1976. More Dematiaceous Hyphamicites. Kew, UK, Commonwealth Mycological Institute. Ellis, M. B., and J. P. Ellis. 1985. Microfungi on Land Plants, an Identification Handbook. London. Croom Helm. 1988. Microfungi on Miscellaneous Substrates, an Identification Handbook. London. Croom Helm. Hughes, S. J. 1976. Sooty Molds Mycology 68:693-820. Ken, J., R. Somervell, L. Sigler, S. Krajden, and G. Land. 1997. Laboratory Handbook of Dermatophytes. Belmont, CA, Star Publications. Kendrick, B. ed. 1979. The Whole Fungus. Volumes 1 and 2. Ottawa, National Museums of Canada. 1989. Subdivision Deuteromycotinae Fungal Chimeracidoia 41:6-14. Kendrick, B. and T. R. Nagraj. 1979. Morphological Terms in Fungi Imperfecti. The Whole Fungus 1:43-61. Kirk, P. M., P. F. Cannon, J. C. David, and J. A. Stolpers. 2001. Dictionary of the Fungi. 9th ed. Wallingford, UK, Cab International. Nagraj, T. R. 1993. Chylomycetes Anamorphs with appendage-bearing canidia. Sydney, Canada. Mycology Publications www.mycolog.com. Nagraj, T.R., and G. Morgan-Jones et al. 1972–1982. Genes Generum Chylomycetum. Fascicles I13. Waterloo, Canada, Department of Biology, University of Waterloo. Seaver, F.J. 1978a. North American Cup Fungi and Operculates. Reprint. Monticello, New York, Lebrecht and Kramer. 19786. North American Cup Fungi Operculates. Reprint. Monticello, New York, Lebrecht and Kramer. Seifert K., G. Morgan-Jones, W. Garnes, and B. Kendrick. 2011. The Genera of Hyphamicites. CBS Biodiversity Series 9. Utrecht, Netherlands, CBS Na Fungal Biodiversity Center. Sibainsaan, A. 1984. The Botanicat Askamycetes and their Anamorphs. Vaduz, Germany, Kramer. Sutton, B.C. 1980. The Chylomycetes. Kew, UK, Commonwealth Mycological Institute. Wingfield, M.J., K.A. Seifert, and J.F. Weber, eds. 1993. Ceratocystis and Ophiostoma, Taxonomy. Ecology and Pathogenicity. St. Paul, Minnesota, APS Press. Zheng, R.U. Yang. 1985. Genera of the Ericifaceae Mycotoxin 22:209-63. A few relevant websites. http://www.mycolog.com/chap4a.htm. http://www.mycolog.com/chap4b.htm. Rogers, A. Unraveling the Mystery of the Canadian Whiskey Fungus. http://www.wired.com/2011/05/ff_angels_share/ The Fascinating Story of Bodoinia, a conspicuous mold with a taste for alcohol, that was named only in 2007. 5. Kingdom Eumycota, True Fungi,